Ready to go? All right, so good morning everybody, and welcome to this GIAN series, GIAN lecture series. We're going to have lecture 6 on combining dual receptors for enhanced sensitivity. First of all, brave effort all of you making it to the auditorium despite the inclement weather that you have outside. You might find my voice a little bit cracking today. It's just because I've got a bit of a sore throat, not still used to AC and fan running at the same time. But anyway, I hope I'm able to explain what I would like to explain as part of this dual receptor strategy that I want to present today. So as I promised yesterday, this is going to be a very interesting lecture. I'm also kind of giving you some really some very fresh reports, some fresh results in this. Some of this material that I'm presenting today is basically unpublished work as well. So fresh from the press, as they say. I'm going to be presenting this whole lecture series to you, this lecture to you. In terms of course structure, I mean this all has been changed quite a lot. So I think you are probably well updated through the group that we have. So please follow the group. Don't worry about this course structure. We have to move things around to make sure that you have a very pleasant learning experience. In terms of suggested reading, there are 2 articles I would like to I would request Khalid to share with you. Both of them should be on the Dropbox folder, and these are basically 2 papers. One of them is on the SARS-CoV-2 virus label-free electrochemical nanohybrid MIP apta sensor, and the other one is one which has been published from my research group on Alzheimer's diagnosis beyond cerebrospinal fluid, which is basically a paper telling you how we could move away from painful lumbar punctures and start doing point-of-care diagnostics in simple samples such as the blood samples. All right. Again, this is using MXene and a redox system and molecular imprinted polymers. The main objectives of this particular lecture is to explain to you the rationale behind the use of dual receptors. You know, we've been talking about biosensing devices. We've been talking about the fact that at the heart of the biosensing devices are your bioreceptors, and the thing that you get with those bioreceptors is you get the desired, I mean you would like them to be very selective, you want them to give you the desired specificity, but then you can play around with this when it comes to the sensitivity, and sensitivity is something that's a very important performance metrics, and you need to focus on that in case you would like to, and this will vary from the application to application. So in the case of glucose, we saw that the concentration are normally in the order of millimoles, which is 10 to the power minus 3 moles. When you look at biomarkers, which are normally seen in blood or in the cerebrospinal fluid or the skin interstitial fluid, the concentrations might be very, very low. So the idea is to develop biosensors which can give you the desired dynamic range, which can give you the desired sensitivity as well, where the limit of detection of your biosensors should be able to detect the range which you are normally focusing on. For example, in glucose, you're trying to develop biosensors which can give you a dynamic range of 0 to 20 millimolars, because that's the concentration of glucose, and that's the physiological concentration that you normally you normally come across in the human body. But the biomarkers, they are definitely not going to be very high in concentration. They would be something as low as picograms per milliliter concentrations. So you need to adjust, you need to monitor, you need to develop your biosensing device, and there, as I said, the core there is the receptor, and then how do we change the receptor, how do we make it much more stronger to kind of cover and make it more sensitive, make it ultra sensitive. That's what we're going to understand from this particular lecture here. We're going to be looking at the integration of dual recognition systems, basically dual bioreceptors, and I'm going to give you 3 good examples. MXene, PDA, MIP sensors, that's the paper that I've already put on the, I showed you before. Some work on highly porous gold and polyphenol red and polypyridine based MIP sensors, and most important, as I said, red hot from the press is going to be some results freshly produced in the last week by my research group on aptamer, combining them with the nano MIPs, and again having the complex system there. And then we're going to look at the discussion, and we're going to discuss on the implications. Hopefully you will understand how using dual receptors allows you to increase the sensitivity, selectivity, and how this will impact the translational potential of your product. Okay, so first of all, we ask ourselves the question, why do we need dual receptors, or why can't we manage just with one receptor? Well, sometimes when you have single bioreceptors such as antibodies, aptamers, or just MIPs alone, you have limitations. You can have limitations in terms of stability. Your bioreceptor may not be stable enough. The other problem you have, the other limitation you have is the selectivity. You're not getting the desired selectivity. And the third problem is the signal to noise ratio. You may not be getting the required signal to noise ratio. You may not be getting ultra sensitive detection for the dynamic range that you're aiming towards. So you then employ dual systems, because then they allow in improving the binding affinity, right? The stronger they bind to your target analyte, to your biomarker of interest, the better results you will get. And it also reduces cross reactivity, and it enhances signal amplification. So that's why we employ dual receptor systems. Now we are all scientists, right? We would like to see some evidence here. So first of all, let's have a look at what are the possible combinations that you can have for the dual receptor system. The possible combinations you can have a natural antibody, and you can combine them with an aptamer. You will come across different terms these days being employed: nanobodies, apta sensors, apta bodies, and stuff like that. When you combine an antibody with an aptamer, it becomes an apta body. Similarly, you can combine a MIP with an aptamer. You can combine a MIP with a redox active polymer. Again, this is a question mark here. And then you can also have nanomaterial systems such as MXenes, which are two-dimensional structures, two-dimensional systems. You can use those scaffolds and then combine them with MIPs or enzymes. And I'm going to be giving you some examples there. So again, coming back to this question, why do we need dual receptor systems? We are all scientists. We all believe in evidence. We don't believe in things happening by chance. So here's the evidence on your screen. Apologies for the small font here. This is an example. This particular column is that on dual MIP aptamer sensor systems, wherein you combine two bioreceptors together. This is your target analyte. This is your detection mode. And then on the right hand side, you have limit of detection when you just have the single bioreceptor. And then what happens when you combine the two bioreceptors, and the DOIs, the slides will be shared with you. The DOIs are where you can pull the papers out and you can read them individually, depending on if the biomarker is of interest for you or not. All right. So let's have a look. What happens when you combine aptamers with MIP hybrid? This is again looking at the PSA. Anybody here knows PSA? It's prostate specific antigen. This is a cancer biomarker for prostate cancer, right? Very, very important. It's normally measured electrochemically using electrical impedance spectroscopy. Now, when you have a single bioreceptor, your limit of detection is around 3 picograms per milliliter when you just use the aptamer. However, when you combine the aptamer with a MIP system and make it into a dual bioreceptor system, your limit of detection goes down to 1 picogram per milliliter. Now, these things make a huge difference when you have clinical samples. Your median value could be somewhere around 1.5, 1.8, right? So going down from 3 picograms per milliliter to 1 picogram per milliliter is definitely very useful. If you want to have a look at this paper, you have the DOI on the screen here on the right hand side. Let's have a look at the second evidence that we have here. What happens when you combine aptamer with a MIP on cuprous oxide nanocubes, which is basically a nanoparticle system. And this is for a particular mycotoxin, which is your aflatoxin B1. Again, you're using electrochemical impedance as the detection mode here. What you see here is from 0.9 picograms per milliliter using the aptamer alone, you're able to push down the limit of detection to 0.012, roughly about, I would say roughly about 8 times better sensitivity here, right? So what's happening here? Combining the aptamer with the MIP is giving you an enhanced limit of is giving you an enhanced sensitivity and giving you a lower limit of detection. You're able to push it down. And again, the DOI for this is mentioned on the right hand side here. The third one here is an example where an aptamer has been combined again with another MIP nanohybrid system. It's mainly used for SARS-CoV-2. And this is one of the papers that I've given you as suggested readings, because what you can see here, the technique is the same, electrical impedance spectroscopy. You are able to look at 9 into 10 to the power of 3, which is roughly about 9,000 PFU per milliliter, which is just with the aptamer. From here, from 9,000, you're going down to 3.3. Can you imagine the amount of, how much, by how many orders? Nearly 3 orders, nearly 1,000 times, or nearly 3,000 times increase in sensitivity. This is a fantastic piece of paper, fantastic piece of work that has been published. Again, I think it's Microchemica Acta, which is the journal in which this has been published. Have a look at this paper as well. Another example for dopamine, alone with the aptamer, you're getting 63 namoles, but as soon as you use a MIP system there, again with a nanoparticle system, you have 47 nanomoles sensitivity there. And again, this paper is reported here. And the last example you have is that of insulin. Insulin, as we all know, is a peptide hormone. Alone with the aptamer, you're getting a sensitivity of, or you're getting a limit of detection of 1 nanogram per milliliter. and when you use this with a MIP and a magnetic nanoprobe, you're able to get this, you're able to get a limit of detection of 0.5 nanograms per milliliter. You've almost doubled it, right? So this is, you're making your systems much more sensitive. So these are the evidences which encouraged us as a research group to look forward into dual receptors, and I'm talking about the project that we have, which is the UK Japan MRC AMED project on early diagnosis of Alzheimer's. Again, there the concentration levels we are looking at are picograms per milliliter kind of concentrations, which is picograms is 10 to the power minus 12, right? Just to put things in the right order. Nanograms is 10 to the power minus 9. So I hope the scales are, because I do appreciate that we have a wide audience, we have people from different backgrounds. So just to kind of make sure that everybody understands the units that we are going to be using here. Okay, let's have a look at case study 1. This is again from my research group, which is on amexine MIP PDA sensor. Basically, we've used amexine here with polydopamine, and the target here is small molecule analytes. In this particular case, we are doing tau 441 protein, which is a protein with 441 amino acids. The nice thing with this particular system is you have a high surface density of the amexine, you have the PDA, which is polydopamine, which provides the functional groups so that the MIPS can anchor to that, and you have an improved sensitivity. That's the overall summary. Let's have a look at how this has been done in the group, and this has been published as well, so you can please feel free to read this paper. It's a part of your suggested readings. Your amexine based redox systems and molecular imprinted polymers. So we started off by just using, in this particular case I'm giving you an example wherein you have you're just working with one single bioreceptor, which is going to be MIP in this particular case. There's nothing else here. How do we do the MIP formation? You have your screen printed electrode. The screen printed electrode has been cleaned and rinsed with water, and on that you normally electropolymerize polyaniline, and this polyaniline is polymerized in the presence of your protein, which is your tau 441 protein here, and then you bring in the oxalic acid and you remove, as I think I've already explained this, how the molecular imprinted polymers work, and you remove that with oxalic acid, you remove the template, and then you finally have a templated molecular imprinted polymer to which you would expect your 441 molecules, which are your target biomarkers, to come and bind to that, right? That's happening here. Results look good, and as you can see here, we're looking at femtograms per milliliter concentration here, right? I think it's not very clear here. I think I should be clear in the next slide here. Yeah, so if you look here, yes, so if you look at this concentration here, you're looking at femtograms per milliliter concentrations that you're normally getting here, and this is normally done, these studies were done in PBS, right? This is what everybody does, and in artificial ISF as well. The idea of doing it in artificial ISF is these are in vitro studies, so that you can use these as wearable sensors as well, where you're going to be monitoring in the skin interstitial fluid. But that's if you have the evidence that your biomarker of interest are actually changing over time with and they're changing rapidly, so that they really require continuous monitoring as well. Okay, so I mentioned, and then in the next step what we did was we did this just alone with the MIPS, with the molecular imprinted polymer as the bioreceptor, and then we combined our molecular imprinted bioreceptors or molecular imprinted polymers with the amexines. Now these amexines are basically two-dimensional materials, right? They are very layered kind of structures there. They are nanomaterial systems, and they basically consist of a transition metal, and they normally have carbides of transition metals normally in these there. And these amexines have been seen in many different applications. They've been used for sensors, they've been used for biomedicine, electromagnetic interface shielding, they've been used for electronics as well, your 5G antennas, your electronics, and your smart textiles. They're used for energy storage, they're used for environmental applications, and also for optoelectronic applications as well. So nice, this is one of those two-dimensional materials that my postdoc had already some experience of working with. He knew how to handle it, so he decided to introduce a nanomaterial system to add it to the existing molecular imprinted polymers that we had made out of polyaniline here. So these are some results here. As you can see here, you have the screen printed gold electrodes. You take the screen printed gold electrodes, and then which has got your PDA, and you've also got your molecular imprinted polymers, and then you actually assemble that on top of your vanadium based amexine system that you have. And as a result of that, we see a great enhancement in the sensitivity. We're looking at log concentrations here, so we are able to increase the sensitivity, and we are able to do much more better we're getting much better linearity. If you look here, you're going from nanograms per milliliter to femtograms per milliliter. So what you've done is you've increased the dynamic range here, right? So earlier you were only getting you're only able to detect in the lower region. You're only able to detect in the femtogram per milliliter region. Now here, by combining the two bioreceptors, or in fact combining one bioreceptor with a nanomaterial system, you can show that you've increased the dynamic range. So you can use this particular system not just for early diagnosis, but you can also use it for prognosis, right? People who already are suffering from the disease are undergoing some form of a chemical intervention, some form of a therapeutic intervention. Now, how is the disease progressing, right? How do you find out that? Because obviously people who are suffering from Alzheimer's disease will have a much higher concentration of the biomarker, right? So if you just are able to do early diagnosis, that's fine, but what you would also like to do is also do a prognosis of what's going on. How is the disease progressing? Do you have sensors which can tell how is the body reacting in response to the drug therapy that's going on there, right? And these days we all know there are a lot of drugs that have been introduced for treating and managing Alzheimer's disease as well, which can slow down your neural degradation. But again, as I said, you need the evidence, and that evidence can only come through prognosis, through prognostic screening, right? You keep on monitoring the levels and see, okay, the person is on drug, is he reacting to the drug or not? Should we increase the drug or not? So you need not just early diagnosis, you also need prognosis. You also need to be able to follow the disease through the biosensing or through the diagnostic devices that you normally have. So that was the case study, or that was the example of the first one, which is the MXene based redox system and molecular imprinted polymer. Now we move to the next one, which is basically an example of highly porous gold with polyphenol red and polypyridine based MIP sensors. The idea again here is the same. MXenes have got a problem. MXenes have got transition metals, right? When you have something which is transition metals, there's always a bit of reluctance there, that even if that nanomaterial system isn't going inside your body, you still have some reluctance even if it's coming in contact with capillary blood, right? So what do you do? And the other problem is MXene doesn't have very good anti-fouling properties. Just even when you drop even when you put a drop of blood on an electrode, you will see some form of biofouling, which is basically the proteins in the blood would go and adsorb on the surface of the sensor. So you should be able to do measurements without any interference from the proteins in your blood samples. So what we did was, after we saw that MXenes was, we did our first work when we were at Swansea University on the use of MXenes, and then when we moved to University of Liverpool, we decided to change the design, the architecture of the sensor, and we said, let's make something which is ultra sensitive, which gives you the desired dynamic range for monitoring of Alzheimer's disease, but based on something which is more biocompatible. And that's when we came across this particular material, which is called as highly porous gold, right? HPG. And we combine this with a MIP system, which is basically a polyphenol red based MIP system, and which has also got some polypyrrole also in it. And as I said, as you can see from the image, the idea is that you have a person who doesn't have to go to a specialized hospital, right? If you're talking about Alzheimer's, right, the gold standard for Alzheimer's, as I might have already mentioned, is basically wherein a patient goes into a specialized hospital, you get the highest concentration of the biomarkers of Alzheimer's in, louder? CSF, cerebrospinal fluid. How do you extract the cerebrospinal fluid? Through lumbar puncture, right? And lumbar puncture it's a very invasive method, right? So until we have pioneers working in the area of sampling methods, give us something easier to get the cerebrospinal fluid, it's going to be a very painful method, and you need a specialized person for that, because you basically have a thick needle pushed into your through your vertebra into your spinal cord to extract the cerebrospinal fluid there. Right? Not a very pleasant experience. You can damage a nerve, right, in the whole process of trying to access the fluid and stuff like that. So therefore what we want to do is move away from cerebrospinal fluid into something that's easily accessible, right? What is easily accessible? Okay, I know the first answer would be skin interstitial fluid or sweat, right? But sweat doesn't have the biomarker. So what is our next best available by sample matrix? Then blood. Which blood? Venous blood, is that more easily available? Capillary blood, because people are used to pricking their fingers to draw capillary blood. So if we can do measurements in capillary blood, or for that matter, even if you went to a GP practice, if you went to see your physician there, and if he can easily draw some venous blood, it's not rocket science. I mean, he just needs to puncture your vein and just draw a little bit of blood there. I mean, I know they have this practice of doing 5 milliliters so that they can do all kinds of tests on you, but you know, you need a very small amount of blood there. This is a very simple, this cartoon basically is showing a very simple case thing wherein you are able to do all these measurements in capillary blood, right? And we are actually very interested in capillary blood as well. Okay, so this is basically a representation of how the work was done. We are basically taking capillary blood and we are using our what we call as the PCB electrodes that you see here, which has got 2 different working electrodes, 1 counter electrode and 1 reference electrode, and we basically these has been functionalized with your nanoparticle system and your MIPS on top of it, and then we are using all the data that comes in there. We do a lot of clinical studies, and then we use our machine learning regressor tree, and these are things that you'll have to speak to my PhD student about, because this is all his business. And then from that data analytics, we try to predict actual values as well. So you need to train your algorithms, machine learning, and get your AI tools there, which then help in predicting. The reason we also need to bring in machine learning and AI, I would just spend a few moments to explain that as well. Now what happens is you have the biomarker levels, right? So you can say the biomarker level is at the median value is around 10. But in a healthy person who's kind of hitting the age of 80, the levels could be 100, 106. So you need to do a correlation with the age as well. Therefore, you need to bring in the different tools which can, if you know that there is a trend there that the biomarker levels will increase with age, and it could mean that the person has got high biomarker levels, but the person is he's a control person. He doesn't have AD there. And that can be ascertained through cognitive tests, right? I'm talking about biosensing devices here, but remember one thing, in clinical situations, if we have a clinician amongst us, that clinician will say you that it's not based on one parameter. You look at multiple parameters. You look at cognitive tests, you look at different biomarkers, and that's why we talk about multi-analyte sensing. Very, very important. Multi-analyte biosensing. You look at multiple analytes. If you have the amyloid proteins, you don't look at just amyloid beta, A beta 40. You look at the ratio of A beta 40 and A beta 42. One of that will be overexpressed, one of that will be underexpressed, right? You look at that particular ratio, you get a number for that. You look at the value of the NFL, you look at the value of the p-tau proteins. So you have multiple analytes, and then you look at the cognitive tests as well. And then you make a decision, how is the disease, number one, so you have to do patient stratification. You need to ascertain, like in cancer, you have different stages, right, based on the severity of the cancer. Similarly, in all the diseases, you have a certain parameter, that you have a certain standardization of how you classify them according to the severity, right? So you need all the tools that you can get. You need multi-analyte biosensors so that you can get all the information you need to ascertain how severe the case of Alzheimer's disease is. Right? And therefore, you not only need your biosensing devices, you also need your machine learning tools, you need your AI, so that you can build what we call as a clinical decision support system, a CDSS, right? That's very, very important. Okay, so let's have a look at what happened. What did we do in this particular case here? So here again, the system has got highly porous gold. We're using polyphenol red and polypyrrole and an MIP. Now, one of the things that I forgot to mention before when we did the first case study is, when we had this particular system here, what we also had to do is, to be able to read, we had to do electrical impedance spectroscopy and differential pulse voltammetry, but we had to bring in a redox probe. We would bring in a solution of Fe2, Fe3 plus, so that we can basically look at the impedance measurements there, right? So we needed an external redox probe to do the readings, to look at the surface. So as I said, what is happening is you have the surface. So what you have is, you have the metal surface. On the metal surface, you have your nanoparticle system. On the nanoparticle system, you have your polymer. This molecular imprinted polymer has got your cavities, right? And just to kind of, so when you measure the impedance here, you got cavities coming directly in contact with your nanoparticle system, coming in contact with your metal surface, right? So if the electron transfer is going to be very easy. If you have a solution, so you bring in a solution of Fe2 plus, so you can easily see the oxidation and reduction. There's very low impedance there, but the signal starts to drop as you have binding of molecules onto the sites. So this now has now increased the impedance, And as you keep on binding more and more molecules to the sites, you then, what you're basically doing is you're increasing the resistance, you're increasing the impedance there, right? And that's normally measured in the form of a signal drop, right? So you see the signal loss thing that we see here, that's basically how the signal loss is seen. And the nice thing is this signal loss nicely correlates to the concentration here. So in our first work, we were using an external redox probe on top of the whole system that we had, and we wanted to move away from that. So we used polyphenol red and polypyrrole, which are themselves redox polymers. So now we don't need an external redox probe, if that makes sense. Okay, so we've done this, and what we have is, as I said, these are redox active composite polymers which have got a very high surface area. Getting a very high surface area for highly porous gold. The other thing with highly porous gold is it's got very good anti-fouling properties, right? Nothing sticks to it that much. And then you're basically using machine learning models for classification and quantification. And in terms of performance, you're able to measure as low as 498 or 0.5 500 femtograms per milliliter limit of detection. And there's also this something called as ROC AUC, area under the curve, and ROC is receiver operating control. So normally the good value is anything above 0.8 is considered to be a good value here. Okay, so that was a summary. This is, what is this? Anybody? What does it look like? Highly porous gold. So these are SEM images of highly porous gold. Just to show you exactly, when you zoom in, right, you can see this. So this is the normal gold, and then this is how we have look at the highly porous gold. I think the scale bars are slightly different here, but these are the different types of, so this looks more like a planar gold. These are how we made different types of highly porous gold, and you can see that. So as I said, the advantage of the nanomaterial system is you get a high surface area to volume ratio. Does everybody here understand the meaning of high surface area to volume? I can give you another good example which you can easily relate to. Can anybody tell me that particular part in the body which, if you keep on stretching it, can take you all the way to the moon? DNA. Fine, that's fine. And I said part of the body. DNA is more kind of a molecule. You're talking about molecular level. Pardon? Anything else? Yeah, nerves, neurons, the same thing. Have you heard about microvilli in the intestine, in the small intestine? What are they? They are basically foldings, right? So if I go back and if I try to show, so if this is it's like a tube, right? If you don't have any inner lining, you see this will have a particular surface area. You can increase this surface area if you have the inner lining which has got finger-like projections, right? Why do we have this finger-like projections in this thing? To increase the absorption, right? That high surface area to volume ratio. Within the same volume, you have a much higher surface area, right? That's what happens with highly porous gold as well, right? Within the same planar gold, you have porosity there. You have pores, and you have these kind of granular structures, right? And on top of this, so this is the nanoparticle system. It enhances the sensitivity. It enhances the signal. It amplifies the signal. And on top of this, you have your molecular imprinted polymer. So you're getting the benefits of two different systems. You're getting the benefit of a highly sensitive bioreceptor, which is your molecular imprinted polymer, sitting on a nanoparticle or a nanomaterial system which is giving you the high surface area to volume ratio. So you're basically increasing the density of your binding sites, right? Rather than being plain, it's up and down. So what's happening again, remember the example of microvilli, right? It's up, you're basically having a much high surface area to volume ratio there. These things together allow you to get a much better system there. Okay, so this is your gold, and you can see this whole scale bar is 200 nanometers. So if you look at these particular granules, there's something ranging between 50 to 100 nanometers kind of scale that we are looking at. So we use this, and then this particular system was mainly designed for p-tau 181. When I say p-tau, anybody knows what does the P stand for here? P is phosphorylation. So which site is it phosphorylation? The biomarker tau 441 is basically a protein which has got 441 amino acids. This one, p-tau 181, is basically where the amino acid has been phosphorylated at site 181. That's what gives it the specificity here. All right. So you can see here we use the same system that I mentioned. You have your your planar gold. On top of that you have your HPG, highly porous gold, right? And then on top of that you have your molecular imprinted polymer made up of phenols, polyphenol red and pyrrole. Again, why do we need polyphenol and polypyrrole? I won't go too much into the details. The papers should be coming out soon. We've already communicated these papers. The papers will come out soon, so you'll get all the information that you need. I know that some of you, I don't want to go delve down too much into the basics, but as I said yesterday, you can tune the materials. It depends on what your target analyte is. Your target analyte could be something which is highly hydrophilic or something which is highly hydrophobic. Combining polyphenol red with polypyrrole gives it the required electrical conductivity. It also gives you the required mechanical stability as well. So sometimes, these are molecular imprinted polymers. You want them to maintain the shape, because here your binding depends on the shape, the geometry, the way the whole thing is three-dimensionally arranged, and it also depends on the charge centers there. Because if you remember yesterday when I gave you that lecture on molecular imprinted polymers, I mentioned about the importance of the interaction mechanisms. I mentioned about the forces that allow you polymerize around it. I mentioned about the hydrophobic interactions which will repel, electrostatic interactions which will attract, hydrogen bonding interactions which will, so you want to maintain that whole thing as if it's staying in a native state. So for that, the most important thing you need is your molecular imprinted polymer, the polymer that you're using to template has to have a certain level of stability. And in our case, we are using something which is redox active. So what you don't have to do is, you don't need to add an external redox probe. You can do all the measurements just by looking at the surface characteristics of your design, of your particular device there. You can use differential pulse voltammetry. I mentioned to you already that we have stopped using EIS. We only use EIS when we are investigating the whole formation, but otherwise we normally use differential pulse voltammetry or square wave voltammetry, because they are more gentle on the system. You're not applying an AC up to from the whole range of AC sinusoidal waves that you normally apply in electrical impedance spectroscopy. All right. So then here are the results in front of you. Very happy to show you this. This is basically your tau protein. You can see the one which is the one which is in the red. This is your human plasma. The one which is in the black, this is again human serum. So basically you can measure this, and we've also done some studies in whole blood as well. So if you just draw some blood and if you just transfer it straight to a biosensing device, no, it never happens. I'll tell you that for sure. You might have seen people take blood and then they transfer it into those ampoules, which then does the separation, which does has got those anti-clogging EDTA and stuff like that, and then you do the separation into plasma and serum and things like that. You can do a lot of those things. So normally you would find most of the p-tau distributes itself in plasma. You have a much higher concentration in the plasma, but we've also shown that we can also measure in serum as well. And the limits of, you can see here concentrations. What limits are we looking here? What is the sensitivity here? What's the dynamic range? We're looking from 0 all the way to 25 picograms, right? Is that the right range? How will we find out what is the right range? We look at the graph below, right? This is the violin plot for Alzheimer's disease, and this is a violin plot for non-Alzheimer's here, right? As I said, everybody has got a different way of, everybody's body is different. Everybody expresses biomarkers in a different way, right? This is the median value for a person with Alzheimer's, which is around between 4 and 6, which is around 5 picograms per milliliter. This is that for a non-AD or a controlled sample. This is around roughly about 3, and this is roughly around 5.5, right? This is 5.5 and this is around 3.0, right? Picograms per milliliter. Look at the difference, how subtle the difference is. Now I've given you violin plots here. What are these violin plots for? The violin plots normally say these are based on the studies that have been done on multiple participants, right? So what we've seen is people with Alzheimer's, the median value is, the values range from here all the way till here, right? You can have healthy controls, the values range from something around 1 all the way to about 6. So you can have those outliers as well, as I said, right? So, but here, if 5.5 is the cutoff, this is the median value, and here you're getting a value of roughly 6. But again, this person is still a healthy volunteer. or a control, not an Alzheimer's person. Why are the levels high here? Because this is something related to the age, right? So you need to do the age related correlation as well. These violin plots show you the width of the violin. This width here is actually telling you that majority of the people, you know, how you get the median value, majority of the people have got this value of about 5.5, and this violin plot shows here the same thing in healthy volunteers is around 3. This is where the majority of the things sit here. This is how you read the violin plots. You'll come across these violin plots. Nice way of describing your results, because here you're also factoring all the other, you know, you're not only telling about you know, the range, but you're also telling about the frequency of that range as well. Okay, more people with Alzheimer's disease have got a value of 5.5. That's the median value, right? Because what does a clinician want to know? I mean, if you send it to an analytical chemist, the first thing he'll ask you is, what is the cutoff? At what point do you differentiate between Alzheimer's and non-Alzheimer's, right? And then you say, well, there's nothing like that. There's no one particular value which will help you decide here. You need multiple. That's why I'm trying to stress here, you need multi-analyte sensing. You look at multiple biomarkers, you look at their levels, you literally have a spreadsheet, and then you also have the cognitive tests, and then the last column is age, and you say, oh, the levels are very high, the levels are around this one. In principle, this person should be AD positive, Alzheimer's disease positive, but he is not AD positive. Why? And then you go and look at the age. Oh, this is somebody who is 85 years of age. That's why he's got a high level of p-tau 181 protein in it. All right. We are now working on p-tau 217, which is now a WHO recommended biomarker for Alzheimer's, right? But again, as I said, just working on one biomarker does not help you, and that's why when you do your practicals, look at the design that we're going to give you. We're going to give you some PCB electrodes to work on. Look at those designs and ask the question, why do we have multiple working electrodes? We may not have the potentiostat right now to do all 4 electrodes, but at least what we can do is we can try and see if we can get some good values there. At least we can give you some experience of working with multi-analyte sensors or multi-electrode sensors, right? Okay, so this is the example that I wanted to give. Look at the limit of detection here. This is femtograms per milliliter, and this is picograms per milliliter. What's the difference between these two? Order of 3, right? 1000 times, right? So we are our limit of detection, so we can actually go down much more lower, and we can measure very low concentrations. Have we got the desired dynamic range? We would think so, right? But when we went to Japan and did some joint experiments, as I said, some of the values were around 108 picograms per milliliter. Simple. Can't do anything about that, right? So what do? Extend the dynamic range there. So we are now working on extending the dynamic range from 0 to 100, and anything about 108 we would say definitely positive for Alzheimer's disease, but please check the age to see the correlation there. All right, so that's the second example. Now I give you a third example, which I said is basically on neurofilament light chain, right? This is an example. I think we're okay for time, right? Basically, this is another biomarker for Alzheimer's disease, and I can tell you off the record that from our studies done in the UK, Japan. So what we are doing in the UK, Japan project is we are looking at the correlation of the biomarkers in different sample matrices, right? We are looking at capillary blood, we are looking at venous blood, and we are looking at skin interstitial fluid. All right. Now, in Japan, they have the technology to be able to express Alzheimer's disease, human form of Alzheimer's disease, in mice models. Why is it useful to study mice models? They have a short lifespan, right? You can start looking at the disease progression and then extrapolate it to humans, right? You can start looking at the disease progression in a very narrow period of like maybe 3 months, right? You do a study over 3 months. You have the mice model which has been expressed with the human form of Alzheimer's, and then you look at and you keep on measuring the venous blood samples. You keep taking venous blood samples. You keep taking blood samples from venous blood samples can be taken from rat tail. Capillary blood samples, you can take that from the mice as well, and then you can use microdialysis probes to extract out the interstitial fluid. I wouldn't use microneedles here because microneedles for extraction are not the best because the mice skin is very thin. I mean, I remember doing the microdialysis and I ended up going into the peritoneal cavity. It's so thin. So you need to stay away from that. So we've actually refined microdialysis, and microdialysis. We've now got a very good method of having the microdialysis wherein the rat is very much moving around and you can still sample from the skin interstitial fluid. And one of the nice things that has come out of the whole study, I mean, forget about the interstitial fluid, so I'm not talking about wearable sensors here. One of the nice things that has come out of the studies is that there is a very good correlation of the NFL in capillary blood and in venous blood. Is that useful information? Yes? How many of you say no? No, everybody agrees with me, that's very confusing. Why is it useful information? Louder, please. Do you have a microphone? Because you can? Absolutely. So it makes your sample access much more easier. You can use a lancet and you can get capillary blood. If the same concentration of, let's say, if something is found in the skin interstitial fluid, right, why would you want to go and sample into the blood, right? Because skin interstitial fluid can be accessed in a minimally invasive. If something is found in the sweat, why would you want to go and access the skin interstitial fluid underneath the skin? If something is found in the skin interstitial fluid, which can be easily accessed using minimally invasive techniques, why would you want to start using needles and lancets and go into capillary blood? If something can be found in the capillary blood, why would you want to go and look into the venous blood? Especially if you know that there's a good correlation and you have sensors which are ultra sensitive enough, which are giving you the limit of detection, which are giving you the sensitivity, and you are able to cover the dynamic range, right? So NFL is one of those very good molecules, biomarkers, neurofilament light chain, one of those biomarkers which actually we've seen that in the animal studies that we've done has a good correlation in capillary blood and in venous blood as well. NFL is also a biomarker for Alzheimer's disease. So if you had a list of biomarkers, established list of biomarkers for which you have gold standards, so the gold standard of measuring these whole biomarkers is a technique called as SIMOA. Anybody heard about SIMOA? Yeah, it's single molecule analysis, right? And what does it, what exactly is it? It's basically a glorified ELISA system there, which is working on magnetic beads, right? So you have like something like a CD, and you have your magnetic beads. You trap your it's enzyme linked. You got your enzyme and everything mobilized on it, and you spin it, and then each bead goes into a single reservoir, and then you're looking into single molecule. That's why it's called a single molecule analysis system. Okay, and again, it's an optical system. And do you know how much is one of this gold standard SIMOA kit costing? It's 200K. And when I say 200K, I'm talking about 200K pounds, right? And what we are trying to do as part of the UK Japan project is to replace this with a 20 pound kit, which can be done with electrochemical sensing, right? Why? Because you can make your screen printed electrodes or your PCB electrodes for as less as 9 pence. You can have your potentiostats, which can be made for as less as 15 pounds. And then you just, so the device price would be 20 pounds, and that will replace a SIMOA thing. But for that, we need to have biosensors which are ultra sensitive, which are reliable, which are reproducible, which are accurate, right? So it's a very ambitious target, but it's not impossible. Can be done. If I can't do it, one of you would take over and we should be able to do it. Okay, all right. So coming back to case study 3, number 3, which is on neurofilament light chain. The dynamic range of this is 16.21 to 100 picograms per milliliter. This is what is stated in the literature, right? But the actual range is, I think, it's anything, and this is the unhealthy levels is over 30 picograms per level. So when the concentration goes above 30 picograms, then you say this is where it's 30 picograms per level. 11.5 is actually what we call as the median value for a healthy volunteer. But as I said, you may find samples from a healthy volunteer values of around 108 picograms per milliliter. That's why you use those median values, right? Because there's a huge distribution there. You can have 108 picograms per milliliter concentration as well in healthy volunteers, but who are at a certain age as well, because neuronal degradation is something which happens in the body, right? Keeps on happening, right? And you have to just basically deal with it. How does it happen? This is a nice image. What is this of? It's a neuron, right? And as you can see here, this neuron has got, what is this part called? The axon, right? And what is this part called? More known as cyton, right? Cell body as well, cyton as well. You can call it. Sorry for doing a biology class in the middle of the GIAN course. So you have these axon here, and the axon is actually myelinated. It's got a sheath of myelin, a myelin sheath at some areas. What are these gaps called? Well done. Nodes of Ranvier. You guys have studied biology very well. I wish you had done chemistry as well, that strongly. All right, so you've got these nodes of Ranvier. So when you look at these nodes of Ranvier, that's where you find your neurofilaments here. And this is how a neurofilament normally looks like. If you isolated a neurofilament, it's about 60 nanometers in length, and it's about 10 nanometers in diameter. It's more like a cylinder, right? This neurofilament, as it degrades, the neuronal degradation gives you different biomarkers, NFH, NFM, NFL, and alpha int as well. Now look at the differences there. The biggest fragment is the one which is NFH, which is 200 kilodaltons, NFM, which is 150 kilodaltons, and then you have the alpha, which is 66, and our NFL is 68 kilodaltons, right? That's the unit. That's more kind of a unit that's normally used for biological macromolecules there as well, kilodaltons, right? So it's quite a big kind of a cylindrical kind of a structure, and I think I've got a representation here as well. This green ribbons that you see here are basically your NFL here. So you can see here you've got these different 1, 2, 3, 4 units here. That's how it's, and as I said, it's quite a long. If there's a biomarker, it's quite a big biomarker there. It's got a very odd shape. Normally you would expect to have a globular protein or something which is more spherical. This one is more in the form of a cylinder there, right? And if you look at the image, this is an image which is basically showing you the green ribbons that you see here. That's your basically your NFL, which is your 68 kilodaltons molecule here. So I'm just going to run you past this. This is basically a model of your polyphenol red and your polypyridine, and this is basically your NFL. This one is your NFL, this whole thing. This is your NFL, and this thing that sits here, which looks like a chair, a G quadruplet structure. This is, what is this? Where do you find G quadruplet structures? Which bioreceptors show those kind of structures? Aptamers. Aptamers can take that particular form, right? Remember, aptamers can also give you those kind of hairpin structures. Why do they do that? Because you can have you seen these kind of structures? Complementary hydrogen bonding between the strands, and then you can have your molecule, target molecule, which can be easily trapped between the hairpin like structures, and then when you open it up, when you heat it up, it opens up, and then the target can be released as well. Okay, so this is just to show you some indications, because I know some of you are engineers and you wouldn't be able to appreciate what the size is. Just to remember, we are looking here, our target here, or our biomarker here is something which is quite big, which is about, as I said, did we say 68? Yes, 68 kilodaltons here. So this is 68 kilodaltons of NFL. That's how it's neurofilament light fragment. Okay, so apta sensors for NFL, right? So in Japan, they have managed to do apta sensors. They have managed to identify. They work a lot. So our collaborators in Japan, they work on different bioreceptors. They work on enzymes. They work on antibody fragments, and they also work on aptamers as well, right? So if you go and do a Google search on Tokyo University of Agriculture and Technology, TUAT Japan, and if you look for Professor Ikebukura or Professor Asano or Professor Sugawa, they all work on different bioreceptors, and they're all part of this UK Japan project that we have on developing early diagnostic tools and prognostic tools for Alzheimer's. And these guys have actually come up with this particular aptamer here, which is basically, this is just a model I've used, creating a pi mole, so that you can help you visualize it, right? So this particular aptamer can bind to these red sites of the neurofilament of the NFL protein, right? This, as I said, this whole thing is your NFL, and you have these specific binds here, these sites here to which your aptamer can easily bind, right? So this is again data which is, as I said, unpublished data. So you should consider yourselves very privileged, because I'm sharing some unpublished data. Normally doesn't happen there, and Professor Khalid and Dr. Prasoon will vouch for it that nobody presents unpublished data, but I wanted to give you the best. All right, so what is the nice thing about this G quadruplex aptamer here? The nice thing about this one, this was developed in, as I mentioned, TUAT Japan. You even got the names of the students who were involved in this. I think somebody who made this slide, very enthusiastic. I think at the moment we have Mio and Chinming who are in University of Liverpool, who are visiting us for some joint experiments. Damai has just left. So they have actually done these are the people who have been working on this particular work. They've done the G quadruplex aptamer, which has been developed, and they are now trying to make an electrochemical sensor out of it, which we will call as the apta sensor. And they managed to do that. They're using 4 working electrodes, again using the same chips that you've seen. We call them the Elara chips, which says SSRG on top of that, the Smart Sensing Research Group one. And they've managed to get a dynamic range. I think let's ignore this one. Let's have a look at this dynamic range here. Or even if you look at this dynamic range here, 64.28 picograms per milliliter to 30.3050 picograms per milliliter. Everybody happy with that? No? Why? No, that's nanomoles. Picograms per milliliter, if you multiply the milliliter with 1000 and then you multiply the picograms, that becomes nanomoles, right? Everybody happy with this dynamic range? What are we looking for? Yeah, and ideally, so what should be the dynamic range of your sensor? So what should be the, when I say range, give me the values please. Zero to 60. Somebody said 30. Well, 30 is where it just starts, so you need something a little bit more wider. Glucose goes up to maybe 12, 15, but we normally have sensors which give you a dynamic range of 0 to 40, 0 to 20. Yes, well done. 0 to 100, I will take that. I mean, I'm not the one working in the lab, so I always give, okay, fine, go and make 0 to 100, that's fine. So, and I'll probably invite you to do this sensor to do 0 to 100 picograms per milliliter. Yeah, does everybody agree that the dynamic range should be? Yes, please. Don't stick it into his mouth. Let him speak. He can speak loudly. Come on. Don't be shy. Come on. I mean, look, if you answer, then it encourages me. That means that you're understanding what these lectures are all about.
So
that's what
less than 11.5 means 10 to something more.
10 to 15.
10
but,
to 100 or more.
Okay. Again, I'll take you back to the same. You remember I spoke about early diagnosis and prognosis. You need to not only just be able to diagnose things earlier, you also need to be able to follow up what is happening to the biomarker levels in response to a particular treatment. So you need a bit more wider range. That's why what the range of 0 to 100 picograms per milliliter is the right range, right? We'll have a special award for you. Well done. Okay. Now what's the problem here? What's the problem here? 64.28 to 3050 picograms per milliliter. What's the problem here? Are we getting the desired dynamic range? What did we decide? 0 to 100 picograms per milliliter, right? And we know the median value for a healthy volunteer is 11 picograms per milliliter. Don't go into so much details. When it comes to picograms, 0.1, 0.2, 0.3 would hardly make a difference. You know, it's very hard. We can talk about different things, but if you speak to Rishita here, she will tell you making solutions for 0.2, 0.3 picograms per milliliter solution, not an easy thing. Yeah, does everybody agree? 11 picograms per milliliter is the median value. This is the dynamic range, and this one here, is it covering that range? No, it can be used probably as a prognostic tool. It can be used as a tool to kind of tell you what's happening, how is the AD person responding to a therapy, but it will not give you early diagnosis. And what we want is early diagnosis. Let's start doing early diagnosis. And as I said, there are reports which suggest that if you can get in Alzheimer's disease, that the onset of the symptoms starts 15 years before the actual onset of the, before the disease starts, before you start seeing the cognitive impairments. Right? So you want to have ultra sensitive detection there as well. The problem is when you go for ultra-sensitive detection, if you increase the dynamic range, you lose the sensitivity. That's another problem that you have, right? Okay, so does this warrant having another bioreceptor here? Yeah, all right, well done. So while these people in Japan, while our colleagues, our collaborators in Japan were working on, one second, yeah. While our colleagues in Japan were working on the aptamers at their end, we were working on the molecular imprinted polymers, which was basically, which is neurofilament. And here, as you can see, from this value here, we are looking at something. This is the kind of results we are getting, and this one is basically in PBS. Yeah, pay attention. We do in our in vitro optimization in PBS and in, what's the other sample matrix? So if you were to do design, if you were to test your sensors, we're not doing skin, plasma, right? So this is some results in the PBS, and this is some results in the human plasma here. All right, you see this and you see this. Okay, what I'm going to do is I'm going to show you when you combine these two together, this is what it looks like. So you have the green one is the one in human plasma, and the blue one is in PBS. Which one looks better? Why does PBS look better? Because the linearity range is all the way till here, right? Actually more in this way, something like this, right? And here the linearity range is probably till here. Maybe you can extrapolate it till here. Are we happy with this? Should we be happy with our MIPS? I mean 0 to 120 picograms per milliliter. Should we be happy with our MIPS? Yes, you will take it? I wouldn't. Why? Look at this range here. I don't care how far it goes here. What is my linearity range? And that's very close. What is my desired linearity range? 0 to 100 picograms per milliliter. That's my desired. Okay, I won't get all the way till 100. It's going to be very hard. I know that. But what can I do to get the best results here? And this is all about optimization. And then the best answers you get is when you test them with real clinical samples. So we have a opportunity. So just to explain you might ask me the question, why Alzheimer's? Why are you doing a project on Alzheimer's with a country like Japan? Anybody has got the answer for that? Yes. And aging population you have everywhere. Exactly. The demographic changes, and Japan, they are known for longevity as well. There's a region called Okinawa. Anybody heard about Okinawa? What's the speciality about Okinawa? People live over the age of 100 years, right? As I said, we are all victims of our own success. We've made fantastic medicines and everything, but we still need to address this main core issue is quality of life. Living longer is not the thing. Living a good quality of life is what we need, right? This is when we look at our grandparents and our parents. You can see with age, you can see that things slow down. How do you maintain a good quality of life? How do you maintain to be mobile? That's the main task. So if you have an aging population and if you have a very low birth rate, then you have very, it basically complicates the whole situation, right? This is a problem not just in Japan. This is a problem everywhere. In Japan, for some reason, apparently if you look at the statistics, 1/3 of the female population over the age of 65, 1/3 of the female population over the age of 65 suffers from Alzheimer's or dementia. And dementia is basically 70 to 80% cases of dementia are caused by Alzheimer's. You can have other reasons as well. You can have Parkinson's. These are different things. I'm not going to go into the clinical details of those kind of things. You have Parkinson's. You have different types of neurodegenerative disorders. But I think 70 to 80% of the dementia is mainly because of Alzheimer's. Therefore, if you find something which can predict the Alzheimer's, and now we have medication for Alzheimer's as well. So you need to bring in the treatments early on so that you cannot, I don't think you can cure it, because this is a natural degradation process. This is like diabetes. You cannot cure it, but you can help in managing the situation so that the person can maintain a good quality of life, right? If you go to Japan, if on the tubes, you'll see people sitting down and doing Sudokus, right? Those quiz-like things. That's just to keep yourselves mentally aware, just to have a good cognitive skills, basically. Keep on practicing with your brain, right? That's how it works. Okay, now what I've done here is I've put the two, this is Japan and this is UK, right? This is the work done in Japan, this is the work that's done in UK. Okay, now what I want you to do is to look at the two plots, the two graphs. This is basically APTA sensors in PBS and APTA sensors in human plasma. This is APTA sensors in PBS and APTA sensors in human plasma. I want you to tell me one good thing with this and one bad thing with this. And please feel free to use the performance metrics that were discussed yesterday in the lecture. What's the good thing here? Okay, you're not getting the desired range. One, that's a bad thing. Yeah, we've already discussed that. I don't want anybody to repeat that. What's the good thing that you see here?
It will be more specific.
Well done. Well done. Did you know the answer before you came for this lecture series?
No.
Are you sure?
No.
It looks like you're trained, because this is I was not expecting this answer. Very well done. She deserves a special mention. Fantastic. Very well done.
Thank you.
Absolutely right. You can see from the two plots that your aptamer, which is your bioreceptor here, is able to pick up the target molecule in a similar fashion, both in PBS, which doesn't have any other interferent, and in plasma, right? That shows how selective and how specific it is. Do you see the same thing here? No, there's a difference there. That means that our MIP system is not as selective as the aptamers are. Right? I was hoping that this discussion would go on for another 10 minutes, but thank God you saved me a lot of time there. Right? And this was discussed yesterday in the performance metrics as well. Well done on that. Well done all of you. Actually, if you've got that thought process and it's come to your mind that this is what it is, very, very well done here. So now, doesn't it build a nice case? What if we combine these two together? Because we would like to have the specificity, the selectivity of the APTA sensor and the dynamic range of the nano MIP device here. Again, I keep on mentioning nano MIP. Why are we calling it nano MIP? Louder. Lead by example. It's called a nano MIP because what is it made up of? What are the MIPs assembled on? Go on, louder. Don't be shy to give answers. Come on. Exactly, on highly porous gold. So that highly porous gold is your nanomaterial system, right? That microvilli example that I gave you, which will stick to you for the rest of your life. Every time you think about nanomaterials, you will think about the nanomaterial system you have in your own body. You know, our human body is fantastic, right? You can learn a lot from the human body in terms of, how the science works, how things work in the body, and then you can replicate that. I mean, Professor Khalid used to work for a group which was called as bio-inspired technologies. Technologies inspired from the human body, right? So microvilli, highly porous gold, right? That's your, you can look at the analogy there. All right. So again, if you put these two things together, if you put an APTA sensor and a nanomip system together, you can then get what we call as a much better. Will that happen? Has that happened? Well, from the initial results we've got from our PBS, and I believe these results. Can anybody tell me why I believe these results? Error bars, because I can see error bars here. When I see error bars, I know that this has been actually been done, not been made up there, right? And you can see here, what has happened here, look here. So this is the APTA MIP. So what we've done is we've done two things here. We've combined the aptamer plus the nanomip, right? Which is basically what you do is you take your metal surface, you have your nanoparticle system, your highly porous gold. On that, you immobilize your you immobilize your NFL on top of this. Sorry, it's mainly in the form of rods, right? So let's say these rods, and then you have your, sorry, apologies for rushing. Nobody's asked me where is the aptamer. So you have your surface, your highly porous gold surface, highly porous on top of that, and then you have your aptamer, which is bound to the NFL, right? So you have your aptamer, which I'm representing like this, which is basically like a chair, right, on which I'm sitting, G quadruplex structure, and then on that you have your NFL, right, and then you have your polyphenol and everything, and then you eat away these parts to create cavities which look like this. So that's your, this is your nanosystem, that's your aptamer, and that's your template, and that's your MIP, right? That's the combination there. What we also did was, we did one system where you have the aptamer with the nano MIP, and then we also created some surface, also part of the surface also has got some, so you can see here you got, it's not AMTA, I think it's got something wrong. Apta MIP is basically your aptamer with the nano MIP, and then you also have a aptamer nano MIP, and we also mix this with additional nano MIPs there as well. Why? Because when we just have the apta MIPs, this is the apta MIPs, you can see here it goes all the way till here, you're still getting a good dynamic range till here about 30, but if you want to increase this dynamic range all the way up here, which is up till about 50, right, you need to have more design, you need to design a better system here as well, right. So if you have something which is between 0 to 50 picograms per milliliter for a start, that's a good system. As I said, this is some ongoing work there, right. I'm showing this to you so that just to kind of motivate you, to encourage you to make a case that you really can make a lot with these kind of combined, by combining the bioreceptors here. If you look at this, how many systems do we have here? We have an aptamer, we have a nanosystem, we have a MIP, and then in another one, so you have got 3 different types of systems combined here, out of which you have a dual bioreceptor, which is your aptamer and the MIP, and you have a nanoparticle system here, and here you have a apta nano MIP, so you already have 3 parts here, and then you have a nano MIP here as well, which is 2 parts here. So this is like a 5 component system. So one is a 3 component system, so this one is a 3 component system, and this one is a 5 component system, and you can see you're getting much better results. So just to give you an example as to how you can play with the different components and optimize your systems to get ultra sensitive detection. And the idea here, as I said, is to widen the dynamic range so that you can cover a range. You can cover a range which allows not only early diagnosis but also helps you in prognosis and in being able to predict what's happening, or in being able to actually screen the particular patients and tell what's actually happening there. Is there early signs of diagnosis, early signs of AD there, or and how is a person responding to a particular chemical intervention of chemotherapy or a therapeutic intervention there. Okay, I think that's probably my last slide, just last few slides now. So I've given you 3 case studies there. So you have certain advantages. You can amplify the signal, you also can do multi-analyte capability as well, you can have different types of aptamers, you can do that. You have a much improved biocompatibility, better reproducibility, and you can model this engineering potential as well. Challenges to consider: fabrication complexity. I mean I'm showing you the good results. I'm sure my students have got good days and bad days, like all of you have, right? Some days they get good results. So trying to standardize the whole fabrication is something that's really, really important. But that only happens once you've optimized everything and you know that this is my best possible scenario, or this is my best possible architecture. Interfacial stability could be a bit of a problem there. And then data interpretation, especially using machine learning and integrated systems. I already mentioned this to you, that you can have a very high value and the patient might turn out to be a non-AD patient, right? So you need to bring in machine learning and all those things, all those tools which correlate all the different factors that are involved, which include age and sex as well, because the prevalence is much more higher in female population, right? So 65 plus female population with a value of 60 might be a short case of AD, while in males might be a different case. Things like that we need to be taken into account. And then cost of material, you're adding on the material, right, because you're having multiple layers here, so you're adding on the material, so cost can go slightly higher. In terms of future directions, we are going to be looking at smart dual receptors which are responsive to pH or redox state. We're going to integrate this to wearable platforms. I think we're going to be working soon on insulin-based wearable devices. And then we're going to use One Health diagnostics so that we can use them in animals, environment and environmental samples as well. And then the dual receptor, we need to look at the machine learning tools for this dual receptor ones as well. And that's the summary. I've already mentioned that, just to summarize, your dual receptor biosensors can overcome performance limits of single systems. You can have, I've already shown you the case studies there, and you have new directions towards intelligent, adaptable biosensing platforms. Finally, I'd like to acknowledge my research team in Liverpool and also from Japan. The funding bodies for the funding, MRC and AMED. MRC is in the UK, which is the Medical Research Council, and AMED is the equivalent of the Japanese research funding body there. To all my collaborators and my clinical partners. I think that's probably my last slide, and I'm going to open the floor for any questions that you may have. I know we've run massively out of time, but honestly speaking, we still have plenty of time for questions. I'm here just to encourage you, just to give you some information, and I would really encourage any questions that you may have.
Yep. Any questions?
Yeah.
So in the last slide which you have shown, where the dual receptors sensitivity has increased.
Yes.
So next slide, yeah. So this one on this green dots, I hope so that is a green one or red one, but green dots, there the error bar is less. So what might be the reason as compared to the red one?
I think the problem is one is in PBS.
Okay.
And the other one is in blood.
Okay.
The PBS one seemed to have ions which might be interfering there, while with blood, because it might, and those ions might be masked in blood. I think that's the only reason I can think about. I mean, as I said, these are fresh from the press. These are these are latest results that I had. I think maybe done a couple of days back. So I haven't had a chance to discuss the whole results in details, and I'm not that much worried because, both of them are actually in plasma. Both of them are in plasma. For some reason, basically the error bars just indicate that one system is more stable than the other one. Sorry?
Okay.
Okay, I will rephrase. Sorry, ladies, I'll hold on for 1 second. Both the studies have been done in PBS, not in plasma. Sorry, just a correction here. These studies are from PBS. We'll be able to see much better results when it comes to plasma. And the girls are right. It's actually, you have multiple systems when you're moving from 3 to 5 five component systems. So something here is contributing more to the noise. It could be adsorption of ions on the aptamers. It could be anything. We just have to look into details. I haven't looked into that. I just wanted to present this to show that we are getting better performance once we combine the different components together.
One more question.
Yeah, please.
So when you have shown that this sort of the architecture where the MIPS and these aptamers are combined,
yeah,
right? So now while removing the polymer, you make sure that both the MIPS part and the aptamer are combining to provide more sensitivity or specificity to the sample.
Yeah.
Now in this case, standalone aptamers, is the MIPS is facilitating the aptamer performance more, or if you use only aptamer, then aptamer orientation or its interaction would be reduced, something like that?
So from the initial studies that we've done, MIPS is giving you the desired dynamic range, getting you closer to the dynamic range. So it's giving you the dynamic range there. Aptamers is giving you the desired selectivity.
Okay.
So you are exploiting the properties of both of these together to get a highly selective and a highly sensitive biosensing system. Okay. With a wide dynamic range.
Okay. Okay. Thank you.
You're welcome.
Any more questions? Yeah, so just wanted to ask something very basic. What is the sensing modality? Is it pulse voltammetry?
DPV.
DPV.
Differential pulse voltammetry. I didn't go into details because we already have discussed so much about DPV and things like that. Yeah. And I've already mentioned that I think when we were going through, I mean, we've not changed any, in terms of electrochemical sensing methods, we prefer using differential pulse voltammetry or square wave voltammetry.
Yeah. Okay. So why is it represented like as signal loss? Or just because the...
So that is the same thing what as I said. You have your surface, you have your binding sites. As you keep on binding,
so the sites get...
the sites get clogged, get blocked. So you have a drop in the signal.
Right.
because you're increasing the impedance of the system,
Okay.
of the layers,
Okay. Okay.
right? So the electron transport is getting much more, the electron transport of electrons is getting much more.
So higher the concentration, more sites will be used
Exactly.
up.
The higher the concentration, the more signal loss you will have. And the nice thing here is you're not using an external probe.
Okay.
The polymer itself is redox active.
Yeah. Okay. So just one more question. Amexin, is it biocompatible or have you looked into that?
Again, for me, if somebody says amexins are biocompatible, I mean, I've seen little reports where they say they are biocompatible. It's like osmium polymers,
Okay.
osmium tetroxide, they're biocompatible. I'm not sure.
Right.
How can a transition metal become biocompatible overnight? So the ones that we used was vanadium, although we
Vanadium.
are not using it for wearable sensors. So that's why we moved away from wearable, because we wanted to use, as I said to most, as you know, I've been repeating this. You can easily transfer your protocols for point of care devices, biosensing devices, to wearable devices, because at the end of the day you're doing biosensing in both cases, right? So we wanted to use something which is more biocompatible, and highly porous gold is highly biocompatible as well. I mean, gold,
use it.
silver, I mean, gold is used, gold nanoparticles are used in anti-aging formulations. Silver nanoparticles are used as antibacterials. So we know that they are.
Okay. Thank you. Any more questions? Okay. Thank you very much for the very in-depth and informative session. And you have also shared some.
No, I'm
Data
actually
which
very
is not
glad.
published.
Very good answers, you know, from the students. Well done. Thank you all as well.
Thank you.
As I say, you know, when you sit and you answer the questions, it encourages me to do even better.
Yes. Yeah. Absolutely.
Although I'm not in the best of my health today, but that's still I didn't realize that.
So we'll reconvene again at 10:30.
Yes.
Okay.
10:30?
Yeah.
Okay.
Okay, welcome back to the next session of GIAN for today. So this session is actually focused on machine learning for advanced data interpretation and forecasting in wearable health technologies. So when you are actually making devices for continuous monitoring, wearable devices, then you need to have some sort of an algorithm or some sort of a processing system which cleans the data automatically for you. And let's say you are monitoring certain biomarkers, then this model or this algorithm should also give you the right value of the biomarker. Let's say if you're monitoring glucose, then it should give you the correct value of the glucose. So there are many different challenges when if you are using wearable compared to if you are doing POC, point of care diagnostics, or whether you are doing continuous monitoring. So there are many more challenges in continuous monitoring, because when a person is wearing a device and they are sitting, sleeping, going on with their daily activity, so these activity actually adds noise to the sensor signal. So how do you remove this noise from the sensor signal, isolate the sensor signal that you are interested in? Let's say if we are monitoring glucose, how do we identify the changes in signal which are specific only to glucose and not identify, let's say, those related to movement or any other interferences? So these are a few topics that we are going to cover in today's talk right now. So we will actually split the talk into two parts. The first part is data interpretation, or you can say data pre-processing. So we want to automate the process of pre-processing. So we'll look at what are the system requirements, what are the specifications for data pre-processing that is required, and how we can do that. And then we'll look at forecasting. How can you actually forecast analyte levels in advance? And we will take the case study of glucose forecasting as an example and try to understand from there. So I just wanted to show you why we need data pre-processing. So this is some data that we collected during an in vivo experiment from an animal. So on the y-axis you have the voltage signal, and then x-axis is actually time. Okay, so this the input voltage actually was representing pH. So we were actually measuring pH inside the vagus nerve in response to some injections that was given to hormone injections, CCK injections that were CCK stands for cholecystokinin. It's a gut hormone given to the animal. Okay, so if you look at this signal, we can identify some small bumps here, and these bumps correlate nicely with the injections that we are giving. Okay, so this is something which is done post recording, like once we have done the experiment and we look at it visually, we are able to identify. So if you look at this data, you see there is a linear drift to it. Okay, so it is actually, there is a linear drift to it. So now if we are monitoring pH, and if you can recall from yesterday's lectures, what is the sensing modality that we use for monitoring pH? Potentiometry, right? Potentiometry. So in potentiometry, we are measuring the OCP, open circuit potential, and this OCP has a linear drift to it. Okay, so now if you can think, if you are making a wearable pH sensor, let's say, or an implantable pH sensor which is inside the body, you will get this kind of a signal and you will have some drift here. So you cannot directly interpret the pH just looking at the voltage signal. You have to actually remove the drift, and then you can do some sort of interpretation. So this is one data from one experiment, let's say experiment 1, and this is the data from another experiment, experiment 2. So in experiment 2, these bumps in relation to the injection are much smaller and they are not visible. Okay, so this tells us that okay, we have to have some sort of a processing method that can actually remove this drift, clean up the signal, and so that the device can automatically and correctly estimate the analyte level, let's say in this case pH. So this is after cleaning. So if you look at the yellow plots here, this is after the drift has been removed. So you can see the peaks nicely, and here as well you can see the peaks nicely. And what is interesting here is you can see some sort of a dose response. You see the amplitude of the peak is slightly increasing as we go from injection to injection. This is because we were giving increasing dosage of the hormone, so there is some sort of a dose response to it. So this is this dose response, I mean, even though some bumps were visible here, but this dose response feature was not visible unless you remove the drift from it. Okay, so this kind of so that's why we need to actually do some data pre-processing before we can do any analysis or any interpretation as to the concentration or pH of the data that we are collecting. Okay, so this particular data was collected when the animal was anesthetized and the environment was more or less stable. But if, let's say, imagine if the animal is having the device inside and the animal is moving, it would have been very, very challenging. Okay, so there the processing will be much more difficult. So starting, if you think of one of the challenges, that is the first feature that is present in wearable sensors, like we see here, is actually drift, sensor drift. Okay, so there is always some sort of a sensor drift. So this sensor drift can be linear or it can be non-linear. Okay, so what we see here are examples of mostly linear sensor drift. Now there can be other interference. So this is mostly linear. When we talk about non-linear, so this sensor drift, the trend, if we look at this trend here, mostly it is going down, right? It's going down, but it can also change the direction. What will you do? So you cannot apply a linear fit to it. So you have to actually do something in real time so that you are actually able to distinguish between drift and signal. Okay, so let's say if this in addition to this bump, for example, here it suddenly goes up, this is non-specific, it's not due to injection, and so it's something non-specific, or it could be an artifact that we got while we are actually cleaning the signal. But you can see that there are two ways in which drift can go. It can be linear unidirectional, or it can be non-linear, and of course it can go in either direction. It can be positive, negative. So how do we deal with that? So this is the first problem that we encounter when we develop sensors, wearable sensors. So generally, if you can recall the system diagram that we had previously, so there was an amplifier. It can be a trans impedance amplifier, or it can be a simple OTA, operational transconductance amplifier, that will amplify the signal. Then the second one is the filter. Some basic filtering is required to remove the noise, and then this drift is a very, very low frequency drift or low frequency signal. So it actually escapes. Sometimes it escapes the filter, it passes through and comes out through the ADC. So in that case, actually we have to apply some processing techniques to remove this drift. So I will show you one of the processing techniques. There are many that are there. So of course the features are enhanced after pre-processing, primarily drift removal. So we are not there in AI yet. So we are still in the signal processing domain. Okay, so this is the example of like from where the data was collected. So there was the vagus nerve from which we were collecting the data. So these were actually very small micro spike, that we saw yesterday, pH electrodes. Okay, so these were micro spike pH electrodes that were inserted into the nerve. The nerve is barely visible because it's this is a vagus nerve from a male Wistar rat, and the thickness of this vagus nerve is around 100 to 200 micron. Okay, so it's very thin, like a human hair, if I were to give an analogy, and then inserting a spike through that is very, very challenging, and then again getting the data out of it is another challenge. So this data which I had shown you earlier is actually taken from this experiment where the rat was anesthetized, the stomach was opened, and the subdiaphragmatic vagus nerve was picked out, and the spikes were inserted, and pH was monitored, and we wanted to see if there is any correlation. So this hormone cholecystokinin, CCK, is supposed to elicit some neural responses in the vagus nerve. So we all know that there are some electrical neural responses. Whether this electrical neural response leads to some sort of a pH response or not. That was the experiment that was being done. Okay, so you can see in this kind of scenario, this is, I mean, the future thought process was that you can make a implantable device inside the body, and you can avoid the problems that you encounter with electrical signaling like noise and interference and all this. So, but because pH is a very slow signal and you can easily filter it out. Generally, when you are recording inside the body, let's say you are recording biosignals inside the body, whether it is neural signals, or you can, if you are recording from a certain muscle, what happens is the EMG and the neural activity, they share the same bandwidth. So you cannot actually use filters to remove, you cannot isolate EMG or non-specific activity from the neural activity. So then pH is something which is very, very low frequency and out of the frequency range of the EMG signal, so you can easily isolate it. So that was one of the reasons why we were looking at pH. Anyway, so what we observed here was drift and how to remove drift. So that the next question is how to remove the drift, and this was actually published, this work was published. So drift can be caused due to various reasons. Just have a look at, so we are familiar with this iridium oxide sensors because we will also see in the hands-on demo session, and these are pH sensors, and we also saw it yesterday. So they actually have a problem of drift, and drift can be caused due to various reasons. Change in open circuit potential, it can be due to background activity, some other activity is affecting the pH, or some sort of a movement, noise, anything. It can be, so if you have a amplifier, if you are using an amplifier with a input bias current in the range of picoamps also, that can also lead to drift. Okay, so I told you yesterday that we should go for something which is in the range of femtoamps, very small input bias current. So generally OCP drift is unidirectional, but it can also be bidirectional depending upon if you are combining multiple causes of the OCP together, then it can be bidirectional also. And this is, so these are some typical recordings that we collected. So it's very hard to see the peaks here. You can see somewhat of a peak here. These are visible, these are the injection peaks, but you can see with the with the drift, it is very hard to make any interpretation out of it. Okay, only if a person can manually look at it, remove the drift, and do it post recording, then only you can actually see. So we try to develop a method where you can do it in real time. Okay, so how we can do it in real time, we are going to see. So generally when we are collecting signals, the signal here, so we are actually, if you are observing signals in the computer, so we are actually converting analog signals into digital signals. So this pH is also an analog signal, and this analog signal generally when we do the sampling, we do at a very high rate of sampling, that's 10 kilohertz, 20 kilohertz, depending upon the bandwidth of the signal. There is something called as Nyquist frequency. So Nyquist frequency is Nyquist criteria, that your sampling frequency should be at least twice the signal frequency. So generally we want to satisfy the Nyquist frequency and we sample at a very high rate. So what we have to do is now for a pH, actually it is moving very slowly, the signal is changing very slowly. So bandwidth tells us, actually just to give you a background, how fast a signal is changing with respect to time. So if a signal is changing very fast, then we say that it's a, with respect to time, then we say that it is a very high frequency signal. If the signal is slowly changing, then we call it as a low frequency signal. For example, you know, all these power supply, AC power supply, they are 50 hertz. So that means the signal is changing 50 times in 1 second. Okay, so that is what we are indicating. So pH is a very low frequency signal. Similarly, glucose also, I will come to glucose reading. We generally take a sample every 5 minutes. Generally all the CGM sensors, they take a reading every 5 minutes, but internally they are sampling at a higher rate because the electronics is designed for higher sampling rates. And this is the pH signal. You can look at the voltages, different voltages here. So this is the kind of peak that we want to detect. So this is in relation to, this is the change in pH that happens due to an injection, and that we want to actually detect. So this actually is inverted because the previous one is actually, if we go back to the previous slide, so this is a voltage in voltage signal, and the sensitivity is generally, as we saw yesterday, it is minus 0.075 millivolt per pH. Okay, so when you convert it into pH, so the signal is inverted. Okay, So that's why we have, so if the voltage is increasing, that means the pH is going down. So that's so this signal generally, this what we are showing here is after drift removal, and so it's easy to look at the dose response. So this is the dose response curve. So with respect to different dosage of CCK, 50, 100, 300, 1000 picomoles per kg, you can see that there are different responses or changes in pH. Now this is done post experiment. Okay, so how do you do it in real time? That is the bigger question, because you want to have a device which will do it in real time. So this is one approach in which we have actually used previously. So we used actually a CIC filter. So the CIC filter actually reduces the sampling rate from 20 kilohertz to around 5 hertz. You can actually program. So this CIC is called as a cascaded integrator COM filter. They are generally used for reducing the sampling rate from a very high sample rate to lower sampling rate without sacrificing the signal quality. So CIC filter is doing that. And then we have an algorithm, a method for drift reduction. So you first detect the drift and then you subtract it. So I'll show you how it is done. It's very simple. So this is the CIC filter structure. I'm not going into detail of that, but if you look at the this is the you can say the mathematical approach to remove the drift. So you look at the present sample, subtract it from the past output, and then you can generate the present input with the combination of the past and the present output. Okay, so this simple mathematical formula, it can help you remove drift in real time. Okay, so if you are, it can be applied to any other biosensors also. You can apply it to this method. So this is called as a correction factor. So this CF factor controls what is the contribution of this term to the overall mathematical expression. Okay, so by controlling this CF, you can actually control how much drift you want to remove. So you can do some sort of a correction on that. So that is one approach. That this is a pre-processing method. No AI involved here. It's simply the mathematical model that can be used to do it. And here, so it works quite well. Yes. So it works quite well, but the problem arises when we have lot of non-specific changes. So in the previous signal, we did not have many non-specific activity, mostly linear in one direction or the other direction. But let's say you have lot of spikes, so it's very hard to actually distinguish between these. So that is where we will be using AI. So if you have, so generally you have this kind of activity, right? We have a linear, very slow changing drift, and then we have some changes in the pH. So the slope of, so this, if let's say this is specific to injection, this is changing at a faster rate compared to this. Okay, so this method is applicable there. But if let's say this drift is also changing at the same rate as the pH, then it will be difficult to remove. Okay, so we'll look at a much more advanced method where if your signal, if your background signal or the interfering signal is changing at the same rate as the signal that you are interested in, what method to use. So you cannot use this method. So this method is only applicable for non-linear drift removal if the rate of change of the non-specific signal is different or lower than the rate of change of the signal in interest. Okay, so this will be actually coming to the next step here. So we use this method to successfully clean it. So now I'll show you some methods which are AI based method, and you can actually use it to do outlier detection. So you can actually use it, let's say you have a scenario where your background is also changing at the same rate as the signal that you are interested in. You can still do outlier detection. You can still isolate them. Okay, so how to isolate that? This is the method which I am going to show now. So this was actually published in a journal paper, and they actually made a chip out of it, like a ASIC, application specific integrated circuit, which I was talking about yesterday. So let's see how they are doing it. Okay, so the way they are doing it is they are actually using a method called PCR. So PCR stands for principal component regression. Okay, principal component regression. So this PCR is a method that is used in, there is a field of study called chemometrics. So there actually in chemometrics, PCR is used quite frequently to estimate the concentration of an analyte from a signal. Okay. So here in this particular paper, they are actually trying to measure dopamine in the brain with respect to stimulation. Okay. So if you stimulate certain areas of the brain, and there will be some dopamine release. Now the dopamine sensor actually that they are using, it shows, you know, it shows responses due to dopamine. So this CDA is the response due to dopamine. It also shows changes due to background. It can also show changes if, let's say, there is a change in pH in and around the brain or in the environment. So what they did was actually they used this PCR to isolate and also estimate the concentration of dopamine from the combined signal. So in the combined signal, you will have changes due to background, you may have changes due to pH, may or may not, but they have considered it, and then you may have changes due to dopamine. But using PCR, you can isolate this dopamine change and you can actually calculate the concentration of the dopamine. So the way they are doing it is, so if you look at this, they have a calibration step where they are manually, they are having the sensor, they have a fluid channel and they are actually putting the sensor in the fluid channel and they are manually changing each analyte separately. Okay. And they are generating data sets for background, so the non-specific background signal. They are generating data sets with respect to dopamine, controlled concentrations of dopamine, and they are also doing by changing the pH. In this way, they generate a data set, and this is their training set actually. This is actually, they are actually using a method called as PCA. PCA is an you can say unsupervised approach where you are actually there isn't a training step as such involved, but I will explain what it is. So these are the different data sets that they are getting for only dopamine, only pH and only background, and then they are making this data set A, and then they are applying this SVD. SVD stands for single value decomposition. Okay, single value decomposition. So using this single value decomposition, they are actually able to generate this matrix. There are 3 different matrices that is being generated. So out of these, one of these matrices is called as the PCs, principal components. Okay. So these principal components, using this principal component, you can actually generate a projection of, you know, if you have a signal which is coming in mixed with background, and you generate a projection only for dopamine, then you can see how similar this only dopamine projection is with respect to the mixed signal. So then you can find out what is the contribution of the dopamine only in the mixed signal. Okay. So that is the approach that they are using. So using this principal component analysis, they are actually able to isolate the contribution of only dopamine, only background, only pH, and then when they have a real time signal coming in, in real time when they are monitoring the brain, they try to match it with the projections for dopamine only, and then from the mixed signal, and by finding out the kind of similarity between the mixed signal and the dopamine only projection, they are able to isolate what is the dopamine contribution to the signal. So they do one more step ahead. The first step is PCA. Okay. So this PCR actually is a combination of, PCR is actually a combination of 2 machine learning methods. The first one is PCA, then the second one is linear regression. Okay. So this PCA will help you isolate the signal which is specific to dopamine, and then using linear regression you can actually estimate the concentration. Okay. So these are 2 different steps. So in combined they are also called as PCR, and this PCR actually is used in so what they do is, when they make the chip, actually they cannot do the training on the chip itself. So what they do is, they do the training of the chip, and then they generate this principal component matrix and the regression matrices that they load into the chip. Okay. And then they can use the chip for concentration. So we also used a similar approach in this similar way. So let's say so this is, you can see here, So the yellow one is the actual data, and then the projected data. Let's say I am only interested in the CCK data. So you can see that the CCK specific signal, if there is any component in the waveform real time data which is matching with the CCK response, then they are overlapping nicely, and the residuals is very low. Residuals means the difference between the two is very low, and when they are not matching, you can see that there is a difference, and by using a threshold on the residuals, you can kind of understand whether the change is due to CCK or not due to CCK, even though if it is linear. So this approach is a way to do non-specific drift removal, or you can do outlier removal using some sort of a machine learning method. Okay, so this is where, so like this, there are actually many different 2-3 methods are there. Actually, you can use non-linear drift for non-linear drift removal, but all these things are done before. So this is done separate from the actual application. Let's say I am going to use it for artificial pancreas. If there is some noise in the sensor, this is the noise removal part or the pre-processing part that is there. Okay, now once I have done it, once I have done the noise removal and pre-processing, now I have to focus on the actual application, which is the glucose forecasting. So just to I have included these slides just to recap few things. So we are done with the interpretation part, data interpretation part. Now we are moving on to the forecasting part, and then in the forecasting we will see how we do the forecasting. First of all, why we do the forecasting. Second is how we do the forecasting, and what are the challenges in forecasting. That's what we are going to look at right now. So as you know, we have been discussing over the past few days the CGM sensors are mostly monitoring from the ISF, and there is a delay, physiological delay between the ISF and the blood glucose level. So you need to forecast in advance so that you can actually stay ahead with the change in glucose levels, and you can do the insulin dosing correctly. Okay, so the AI model actually sits here in the controller, and this controller actually does two things. First, it does the forecasting. Okay, after it does the forecasting, it estimates the insulin dose, and this insulin dose is given to the actuator. So this actuator, the insulin pump, is the actuator. Okay, so in today's session we will only be focusing on forecasting. And these things we have already seen. This is the technology stack that we have. So yesterday we looked at technology stack 1 and 2. So today we are looking primarily at technology stack 3. So we are mostly looking at technology stack 3. Okay, that is the focus of today's lecture, right? So what is involved in this glucose forecasting? So essentially you have the patient or the person who wants the glucose forecast. So you have actually many different factors which are affecting your glucose level. First is the physical activity. So the major part is actually meal and insulin. If they are taking insulin, then these are the two major contributors to change in glucose level. The other one is physical activity. So then physical activity also, there are two types of physical activity. There is aerobic and then there is anaerobic. Okay, so aerobic and anaerobic will actually affect the glucose in different ways. So aerobic will lead to decrease in glucose level, anaerobic may lead to increase in glucose level. Okay, so and this data, this glucose data is collected through CGM sensors, and then we use the there is a physiological delay between the blood glucose level and the CGM level. So we need to do the glucose forecasting so that we can know in advance what is the glucose level is going to be, and then accordingly based on the forecast you can actually give the dosage, right? So there are two important factors that you need to remember when you are doing forecasting. First of all, your forecast has to be accurate or precise, you can say. Why? Because you are doing the insulin dosing based on this forecast. Okay, so if you get the forecast wrong, then the insulin dose will naturally be wrong. Okay, so that's why they have to have high fidelity. The second thing is the second aspect that we need to remember, the second challenge that we need to tackle is time lag. So whenever you are using AI models, deep learning models, so there is because they have lot of parameters, so there might be a lag between when you want the forecast, there might be some delay due to mathematical computations. So you want to have the time lag as low as possible. Okay, so this is one, and this is two. And the third one, which is more of a important and desired feature, especially for doctors, is actually explainability. So if you have worked previously with neural networks, you will know that the neural networks are like a black box. So once they are trained, you don't exactly know why they are forecasting a certain value. Okay, so it will be good to have a network which is actually explainable, which is giving some interpretation to the doctor. Let's say that okay, this is happening because of this reason, the forecast has been given due to this reason. Okay, so this explainability is another factor. So there are three factors that we are going to look at. First is accuracy, then time lag, and then explainability. Now there is also another factor which is coming inside this time lag. Let's say you want to have a model which you can deploy it on a mobile phone. Now if you want to deploy a model on a mobile phone, that means the number of parameters required for computation for that model must be very less, so that you can fit in the memory of the mobile phone. That means naturally the time lag will be less. So the more number of parameters you have, more processing, more time lag. But when you are having this lower number of parameters, you will have to sacrifice for accuracy. So in engineering always there is trade-offs. So you have to make a decision which how to balance these trade-offs. Okay, so there is a need for accuracy, but if you go for very high accuracy, you might have a large number of parameters. Then large number of parameters means large time lag. So you have to find a sweet spot where you can balance all these things. Okay, so let's have a look. So the first framework that we used is a very simple framework. I'll go into the details of the framework little bit in very shortly. So overall you have this system here where your CGM sensor is collecting some data, sending it to an app, then this app goes to a cloud, and then the deep learning model is running in the cloud. For the framework 1, it is running in the cloud, and then it is sending the forecast back to the mobile phone. Okay, for this particular so basically the training is done in the cloud generally, because cloud has very large number of resources and you can use it. Then the trained model can be downloaded into the mobile app, and then you keep on using it. Okay, and then of course based on the forecast you can give some insulin dosing, and when you are actually estimating the glucose, when you are calculating the glucose forecast, you need to consider multiple parameters, at least 4 parameters you need to consider: past CGM values, insulin dosage past, if you have taken any past insulin dosage, what is the meal, and what is the physical activity level. So these are 4 parameters that you need to keep in mind. So there are many data sets actually nowadays. Type 1 diabetes data sets are lot of data sets are available. Ohio T1D is a very popular data set. It has been there for a very long time. It doesn't have massive number of people, as you can see here, around 1 2 3 4 5 6 7 8 9 10 11 12. So they have actually 2 sets, one into 2018 and the other one is 2020. So they have 2 sets. This is the 2018 set and this is the 2020 set, and in total you might have up to maximum around 20 to 30 people in the data set, and they have data for approximately 2 weeks, and they have physical activity data as well. So generally whoever works in this glucose forecasting, they use this Ohio T1DM data set to train their data and actually do the forecasting and test it, and many papers actually use it. Nowadays there are many other data sets have come up. There is one group in Shanghai which has generated a very large number of data, large data set. There is also another website, there is another data set called T1 DXI or DX1 or something like this, T1 DXC I think. So T1 DXC is also a very large data set which has CGM values, meal information, CHO, carbohydrate, that is meal information, and as well as physical activity. And this is available through a website. The website name is Vivli. So Vivli has a good repository of clinical trials, data from clinical trials. It's like a Google for clinical trial data, and you can actually access it by signing different data usage agreements, that you are going to use this data for ethical purposes, not for commercial applications and all these things. So this is how the data looks like. As if you look at it, I think yesterday Sanjiv was also showing very noisy data, and then you have some points where the data is missing, This is, we have chosen a good one to show you here, but there are points where data is also missing, and you have to somehow, you know, incorporate everything together. So we also collected some data. So for validation, we train on the T1D data set, and you can actually test on your own data set as well. And this is how, so we collected using the Freestyle Libre sensor that is available in India, and then we did some trial in the hospital. We have our own app and cloud system to collect the data. So this is a CGM sensor which is implanted on the arm of a volunteer, and generally we put this patch on it. So this is like a waterproof patch that like if somebody is taking a bath, so you can actually it will not damage the sensor. So what happens here is, in India, the Abbott Freestyle Libre sensor that we have, so there are actually two modules here. This is the CGM sensor, and this is another module called as the BlueCon module. Okay, BlueCon. So generally the Abbott sensor that we have in India, and they only do NFC readings, near field communication. So you can only scan when you want. We don't have the Bluetooth version yet. It's supposed to be released in August or September, but we don't have the Bluetooth version yet. So, but we want, ideally we want Bluetooth data. We don't want the user to scan again and again and again. So that's why this BlueCon module actually sits on top of the CGM sensor. It converts NFC to Bluetooth, and you can get the Bluetooth data in your mobile phone. Okay, so in this way actually we collected the data, and so this is the network that we have here. For some reason this is from a different presentation. So this is the data that we have here. So we have, this is the neural network that we initially designed. So in this neural network actually you see what we use the typical, you know, neural networks that are used for time series processing or forecasting, that is LSTM and GRU, but we did some changes to this network. So we observed that if we use a wide deep approach, so where you have wide deep means all the inputs don't go through all the layers, you treat each input differently, because we observed that CGM data requires more processing compared to insulin and physical activity. Then you pass it through, you know, so the number of layers that CGM is passing through is more compared to that of insulin and physical activity and CHO. So and we use the combination of GRU and LSTM, and that actually led to better performance compared to only LSTM or only GRU. So what are the metrics? How will you validate your model? So these are the typical metrics that are there for machine learning models. So these are forecasting, so they are like regression metrics that you can use, root mean squared error or mean absolute error. But in addition, because we are working in CGM, we also use this MARD. So MARD is mean absolute relative difference. So this is what we have also seen earlier, that is being used. This MARD is also used for, if you are developing your own sensor, you can compare it with a blood glucose and you can get an MARD value. So we use the same MARD also to validate the forecasting accuracy as well. So how good is the forecasting compared to the blood glucose level? So you can actually use, so you get some MARD value also. So this, generally the units for this is milligram per deciliter. This is in percentage. Okay, so when we say that the MARD is 6 or 8, it's actually a percent. Okay, so the difference is only 6% or 8%. So I guess, I don't remember exactly, but the requirement is that it should be less than 10% from regulatory agencies. But MARD is, this is one of the parameters. We also use the Clark grid error that we have seen earlier. So you can see here zone A. So if you are, so this is the reference blood glucose level, and here you can have the measured glucose level, or in other words you can have the forecasted glucose level. We can have the forecasted glucose level, and ideally you want all the points to lie in region A. Okay, so region A is where clinically accurate predictions are there, so there is no major risk to the patient. So this is called as the Clark error grid that we have seen earlier, and then we have the Bland-Altman plots also, and we have the violin plots also. We have mostly, you can actually look at the Clark grid error. And then zone B is also good enough, not bad, not the best, but still workable. And zone C and D are the danger areas which may lead to improper decision making or clinical decisions. So ideally, if you are developing a forecasting method, you want your forecasted points to lie in region A, mostly, and some in region B maybe. So let's see how our model is performing. So this is how the forecasting works. So if you look at the real value and the predicted value, they are matching quite well. In previous works, people who have developed a model previously, so they have a problem at the peaks. Sometimes they will work well, the model works well if the data is changing slowly, but if it is changing rapidly, like in the peaks, whether it is up or down, there might be a difference that we managed to solve here using this model. Second, as you can observe here, there is a delay. So you can see that this plot, the black plot is leading with respect to the purple plot, and that is because of the delay in processing or generating the forecast. So there is around one sample delay. One sample is again is equal to 5 minutes. So this CGM is actually sampling at every 5 minutes. So there is a one sample delay between the actual and the predicted value. So not much, but still we would like to reduce it. So we have used different forecasting horizons. So you can forecast 15 minutes in advance, 30 minutes in advance, or 60 minutes in advance. But as you increase the forecasting horizon, you are generating more number of points. Naturally, your computation increases, and naturally your delay also increases. Okay. So this is kind of performance for this is a for RMSE, MA, and MARD values for all of these different T1D subjects and all that. This is the Clark grid error. So if you can look at this Clark grid error, you see some of the points are there in C also, C and D, but majorly most of it are actually in A, region A. And as you increase the forecasting horizon, naturally you get more number of points outside. So there is some risk, but of course there is a limit up to how much points you can actually tolerate here. So ideally, if you have 90% of points within the A and B region, then you are actually okay, 90 to 95%. Now this is another approach that we tried. So it's always better to update the model frequently. So if you use an approach which is train once, then the MARD actually becomes poor as you move on with different data sets. But if you train frequently on the same individual, if you are training frequently, you are updating your model, then it is actually MARD is actually improving. Okay, and this MARD is very good. This is the ideal MARD, not the actual MARD, because this MARD is from the forecasted data, not actually not the real not the sensor. The sensor MARD is around 8, 6 to 8. 6 is very good, but generally it's around 8. So that is different MARD. So this is the forecasted MARD, not the sensor MARD. Of course, this is comparison with the previous work. We are fine with the metrics that we have actually. If you look at the metrics, then we are performing better than the others. The place where we are falling behind is the time lag. So this is 5 minutes, 1 sample delay, 4 sample delay, and the delay increases as you go for larger number of forecasting window. Okay, so now essentially now we want to actually, there are two problems that we want to resolve. Somehow we are performing well in MARD and the other performance like RMSE, root mean squared error, and other metrics there, but ideally we want to have low delay, as well as we want to add some explainability to the model, so or interpretability to the model. So for this actually we tried a new type of network which is called as the neural basis expansion network, and the neural basis expansion networks, they are actually interpretable. So they give you a trend component and a seasonality component. So they tell you exactly why a certain forecast is given and why the forecast is there. But the accuracy is not as great as the previous model. So we try to enhance this by using multi-head attention layer, which is within the. So this is the typical block of a neural basis expansion network. Generally, neural basis expansion, they work with only one input. So you can actually expand it like we have done it by trying to, we modified the neural basis expansion network to use multiple inputs. So at least 4 are there: past CGM, past CHO. CHO is for meal, past insulin data, and past physical activity. And then you actually try to have these are the different. So we have generally in neural basis expansion network, we enhance it by using this multi-head attention layer. Okay, so this multi-head attention layer helps in improving the accuracy quite a lot, the MARD quite a lot, the RMSE quite a lot, but it actually reduces your interpretability to some extent. Okay, so we call this as a partially interpretable model. Partially interpretable, we get some idea of why a certain forecast is given. I'll show you in subsequent slides. But at the same time, because this is a bit of a black box, so it's not completely interpretable as a standalone neural basis expansion network. So in that way, actually, we try to improve the accuracy as well as. So this work is already published, and in the interest of time, as we are moving a little bit fast, so you can actually look at the MARD. Let's go to the comparison where. So yeah, so nice. You can see here the delay is not there. So in the previous one, there was a 5 minutes delay. The delay has disappeared here. They are actually almost overlapping. So in 15 minutes, and in 30 minutes there is a small delay, and in 60 minutes there is actually a delay of only one sample, around 5 minutes if I remember correctly. So the delay is much better than the previous networks, but and at the same time there is also some level of interpretability. So let's see whether there is any improvement in the forecasting accuracy. So if you look at this plot here, then this gives us in some idea that there were less number of points in this C region compared to the previous network which I had shown you, and there is more number of points in the A and B or the safe regions. So A and B are the safe regions, right. So if you look at the comparison also, we get much less time delay as well. So previously, if you remember, it was around 40 and we brought it down to 15 point something. So the time delay has improved, and at the same time the MARD value also is lesser compared to the previous work. So the best was 15.27, now it is 12.15, and this way we have actually improved the MARD as well. So there are many advantages of this network. The first advantage is time lag is less. Second is accuracy is better. It has high fidelity forecasting. Third is there is some level of interpretability also. So this is how actually interpretability looks like. So let's say this is the overall prediction. So the overall prediction is the blue window, that the blue plot that we are getting. So this is the overall forecast. Now you can interpret why this forecast has been achieved, because this forecast is a combination of the seasonality component and the trend component. Okay, seasonality and trend component. So this, when you combine these two components, you are getting this kind of data. So you can say the trend component is much more of a short term change. It captures short term changes, and seasonality is capturing long term changes. So it's actually coming from stock markets, where you have this trend and seasonality ideas, where you look at short term changes as well as long term changes. So it is telling us that okay, the glucose level is increasing here because based on the seasonality component, there is an increase in the seasonality component that is causing the glucose forecast to increase. Okay, so in this way you get some idea, and seasonality is of course the long term component, and you can say that okay, because the long term, so the patient has seen around this time, mostly the patient has seen an increase in the data. That's why it is actually giving you larger forecast compared to the trend. So there is a combination. So it is, you are able to break this component into two parts, the trend and seasonality. Now there is these are some mathematical equations which are actually showing us how the overall model works nicely. So you cannot just fit the multi-head attention layer into the neural basis expansion. You have to do some there are some corrections and some factors to keep in mind, but we are not going into much detail of these factors right now in interest of time, because I want to show you some other interesting application. So these equations also enable us to extract the trend and seasonality visualization of a glucose forecast. So why a certain forecast has been given, and what is the contribution of the trend component, and what is the contribution of the seasonality component. So in this way you have some level of interpretability, and this interpretability actually helps, keeps the doctor happy as well. They know exactly what is working in what way. So you can also look at different layers. So when you are actually so there is a parameter called as flops per G. So this flops per G is a metric that we use to understand how fast computation is done, what is the requirement of the computational requirements of a particular model. Okay, so this model actually has lower flops per G compared to some of the previous models. It's not the best, but still it has comparatively less compared to, let's say, RNN, dilated RNN. So this DRNN stands for dilated RNN, but still we can actually improve somewhat in this region. It is better than TCN also, this another network. So there is some improvement, also better than deep ensemble network. So in this way, if let's say you are developing your own model, so the first parameters, think about this, let's say you are developing your own model. So what are the parameters that you will test or look at? First is the RMSE, MARD, MAE if you like. RMSE, MARD is enough. Then you look at the time lag. Then you look at the processing requirements. So the flops will give you the processing requirements. Now this flop, if you take any processor, let's say you take your Intel processor, the Intel processor will have certain flops. So if your flops is lower than the specified flops, then you can deploy this model on the processor. If it is more, then you cannot deploy. Okay, so there is this kind of simple comparison. So that's why people look at the flops per G as a metric. They do this analysis to understand where they can deploy their model, whether they are able to deploy it or not. Okay, so still we are not able to deploy this model yet on an edge device. So we want to reduce the flops further to be able to deploy on the edge device. When I say deploy on the edge device, I mean training as well as operation on the edge device. So operation can happen in the edge device, but right now we cannot do training. Training requires lots of flops per G. So that's why we want to actually, generally training is done in the cloud. The trained model is downloaded to the app. So that's how the AI driven, most of the AI driven apps are being used. Right. These are some publications related to that. Now coming to the interesting aspect, I had shown you one plot earlier where I showed you that there is a data missing. Okay, so this happens, this is a CGM data for a particular individual. There is actually a missing data point here. Now if you have missing data points, you cannot actually do any sort of forecasting. Okay, so in order to deal with this, we try to use there is something called as generative adversarial networks, GANs. Okay, generative adversarial networks. So these networks were actually developed by someone called as Ian Goodman. It was part of his PhD actually. And these generative adversarial networks are very versatile networks. You can generate images, and here we have adapted that, so you can actually generate human faces. If you go to, if you can Google now also. So if you type this person doesn't exist, there is a website. So this website actually generates unique faces. So it's not the face that is generated is not the face of a real individual. It has been trained on previous data sets and is generating unique faces, and it is very much similar to a human face. So this model that we are using, we are adapting it. From an image to a time series based data. Okay, so we have adapted that to generate the missing data points here, to replace the missing data points. And while doing that, actually the way generative adversarial network works is there is a generator and there is an adversary. So generator generates different points, it generates a series of points. The adversary actually looks at these points and tries to see whether it matches the probability distribution, the desired probability distribution. Okay, let's say you want to generate the face, then it has a certain spatial distribution of pixels. So the adversary actually ensures that it matches the generated points matches the actual. So it actually removes those points which are noise, and it only keeps those points which are actually matching your desired probability distribution. So it's all about matching a certain probability distribution. So here also you can see this is our GAN network, and what we have done also is that there is this discriminator or the adversary. So this discriminator is we have the generator, and then we have the adversary or the discriminator. So this discriminator actually we observed that if you use LSTM for long sequences, you have a better generation, better discriminator, and if you use GRU for short sequences, you have a better, rather than using a single model for all sorts of sequences. Let's say I want to generate 60-minute data, that is a long sequence, then I would go for an LSTM as a discriminator, and otherwise I would go for GRU as a discriminator. So this kind of distinction if you use, then you can see it performs quite well. So here if you look at this plot in the bottom here, so you have the blue plots which is having missing data points, and the green one is actually generating the missing data points. Okay, so it is actually replacing this real data with synthetic data, and then when you are training your AI model, you can use a combination of real and synthetic data and you can train. Okay, so models require a lot of data to train. Much more detailed diagram, nothing much to see here, we have already discussed. So what do we classify as a long sequence and a short sequence? So generally when we say it is a short sequence, we are using GRU. So generally we want, if you want maximum 24 points to be replaced, then we call it as a short sequence, we would go for a GRU discriminator. And if we are having very large number of data points that we want to generate, we use for an LSTM discriminator. Okay. So the way to validate this is, it's not your typical RMSE, root mean squared error, or mean absolute error. We generally use something called as PCA, principal component analysis, and TSNE. So TSNE is another method, it's a dimensionality reduction method. So you look at the spatial distribution of points, the actual points and the synthetic points, so they should overlap. So if they are overlapping in this space, principal component 1 and principal component 2, then it means that you have a good GAN network. If they are not overlapping, visually it can tell you that okay, this GAN network is not good enough to be deployed. Okay, so here if you look at it, it actually matches quite well. And then you can also generate a quantitative value. So this is qualitative, you can look at it. You can also generate a qualitative value which we call as a discriminative score. Okay, so the lower the discriminative score, the better it is for the GAN network. Okay, so I think this is where we are actually replacing. So not only you can generate CGM, let's say you have some missing data points. Generally the insulin and the meal information the user has to manually enter in the app. So if they forget, that data point is missing. So if you know an overall trend, you can replace those missing data points as well, and you can replace that with a very good fidelity, good accuracy actually. And then you can use a combination of these, you can generate both physical activity data, you can also generate lifestyle data, which is the meal information, and this is meal information, and this is the insulin data, and they are actually, you can see get a visual idea of what is the difference between the generated and the actual points for a certain example, for a single subject. So this is the use of generative AI for certain application in diabetes management. Okay. Right. So there was only one previous work which we were able to compare, and we have slightly better result score compared to the previous work in terms of imputing the missing data points. Right. So right now what we have here is, okay, what we have covered broadly in this topic is we require some sort of pre-processing in the sensor, especially if you are using wearable sensors. So we have to account for linear drift, we have to account for non-linear drift, we also have to account for non-linear drift which has a similar profile to the target analyte of interest. So linear and non-linear drift which are not similar to the peaks that we observe, the rate of change, we can easily remove using a simple mathematical model. If they are similar, then we can use an AI based approach, which is this PCR, principal component regression, or you can use simply, if we just want to do qualitative, then you can simply use PCA as well, do a projection, look at the residuals, and if the residuals are very small, then it belongs, and if it doesn't, then it's not due to the target analyte. That is the first that we covered. Then we also looked at AI based data forecasting. They perform better compared to other approaches. So there are other approaches which actually you can estimate the data. You can also use mathematical models, but AI based approach seems to work better. We have also learned that GAN based methods, so generative adversarial network based methods, can be used to replace missing sensor data points as well. So I would like, when the session is over, so you check out this website. This person does not exist, and you would be surprised what the AI can do. Okay, this person does not exist. So it actually generates unique faces, and it's very it's hard to differentiate between actual face and the generated face. So I think that's the end of my slide. If you have any questions, you can ask. Yeah.
Thanks. Nice presentation. Everything is perfectly fine, very much understandable. Just a quick question. You mentioned about time lags.
Yeah.
Now you're talking about time lags which are more related to your processing time lags.
Yes.
How do you account for the physiological time lags?
So that's why we are forecasting at least 15 minutes in advance. So that is the way to account for your physiological time lag. So we have different forecasting horizons. So if you go back here, yeah, we can see here also. So here you have these different forecasting horizons. So you can forecast at least 15 minutes in advance. So that is the bare minimum physical, that is the maximum physiological delay that you have. And then, so generally what you will have is this delay plus the processing, let's say 5 minutes is the processing delay.
But then why have you gone from 15 towards 60? Why haven't you gone from, because I think the range is normally from
Very
four-
small.
Very small.
Very
It's
small,
between
yeah.
4 to 15.
Yes.
So why have you gone the other way of the spectrum? Why haven't you gone?
So it actually, it's a way to stress test your model.
Okay.
So if you can do very well for 1 hour, then you will do very, very, very well for 15 minutes.
Okay, that
That's
makes
it.
sense. Great. And you also mentioned about those different noise and stuff, drift and stuff like
Yeah,
that.
yeah.
And are you also accounting for any movement artifacts that?
So yeah, so movement artifacts are generally very fast changing.
Yeah.
So let's say if I move my hand,
Yeah.
it's like a peak. So it's like a very fast peak compared to, let's say, so this you can remove using a low pass filter.
Okay.
So that filtering is already there, low pass filter. But if you have, let's say, if in case of neural signals, this is a major problem in case of neural signals. Let's say you have EMG as well as you have neural activity, and they share the similar bandwidth, and then it is hard to remove. You have to use this kind of some other approaches to remove that.
Yeah.
Okay. So in sensor signals, generally, let's say we are taking sensor values, let's say every 5 minutes. So what CGM sensors might be doing is they sample at a very high rate, and then every 5 minutes they are averaging the data and giving you an average value. So when you average out, this peak actually disappears.
Yeah. Makes sense.
Yeah.
Okay. The other question I want to ask is a very general one.
Yeah.
I remember having this discussion there, and many times we've been asked to be careful about not using chemometrics.
Yes. That's right.
And classify that as machine learning stroke AI.
Okay.
Like
Yeah.
even and when I was observing your presentation there, you mentioned a lot of those existing methods, traditional methods, which are used with electronic sensors, like removing drifts, having those
Yes.
filters and stuff like that.
Yeah. Yeah.
I mean, do you also have risk that having that fine line between your traditional methods and people classifying even those things as machine learning and AI tools?
Filters generally they don't consider as AI,
Okay.
but this PCA, principal component analysis, although it has been there for a very long time,
Yeah.
this chemometrics is a very old field.
Yeah.
These people nowadays are telling that okay, PCA is part of machine learning.
Yeah.
It's a part of dimensionality reduction. So that's why I mentioned.
So what are your thoughts? Do you think it's part of machine learning, or do you think they should be separated from machine learning?
I think it is a part of machine learning because it is used quite a lot for data analysis applications, sorry, data science, for these applications. I mean, I'm being a little bit partial to the machine learning
I know. I mean,
side.
because I'm having this problem because I come from this traditional field of
Yes.
analytical chemistry,
Yeah.
where people who have been working on chemometrics are very, very, firm that they do not want to
So they
consider.
classify this as a statistical approach.
Exactly.
Yeah.
Yeah. So it's
But-
more statistical rather than actually machine
Yeah,
learning.
actually, that is true. That is the fact because most of the machine learning is a mixture of statistics, probability, and all these things. So yeah, somewhere it falls in
In the gray area.
gray area.
Thank you. Thank you so much.
Thank you.
Sir, my question is, you have shown the data for continuous monitoring. What
Yeah.
if we are taking point basis samples? Like for, I have taken one sample at 1 hour, then 6 hours later, one sample like that, 24-hour cycles. And if my sensor is targeting two analytes or more than two analytes, in that case, the signal will be very much mixed, right? So let's say I am targeting glucose along with lactate and along with some sodium, potassium like that. So in that case,
Signal bandwidth, I would say, let's say you have a glucose sensor, and then glucose sensor also has issues with other interfering agents. Let's say ascorbate or acetaminophen. I'm just trying to rephrase your question and try to understand. So the glucose sensor will give you a signal even though there is no change in glucose, but there is change in, let's say, ascorbate, right? That is your question.
yes, yes.
So how do you distinguish between two?
Yes, yes.
So if you have trained this chemometric, so it's very hard to do. For chemometry, actually, you require at least a set of data points. It's very hard to do it in spot measurement. So this chemometry actually comes from NMR, right? Actually it originates from these NMR curves. So you require a continuous form of data to do. For spot measurements, it will be hard. One thing you can do is sometimes people use differential measurements. Differential measurement means, for example, you have a sensor which is reading your target analyte plus background, and then you have another sensor which is only reading background. So you can subtract this differential. Yeah, exactly.
Any other questions? So I have one question. So you have worked with some clinicians as well, you are showing.
Yeah.
Right. So how much the clinicians have the confidence in the missed data or the data which you are trying to forecast? What's your experience on that?
So my experience is that so clinicians like to understand why what is happening. Like for example, if you add some level of explainability to your AI model, so some clinicians, or at least those who have worked with, they are open to using it. Okay, so it leads to the question is whether it is widely adopted or not. So that still we are actually going through a phase of. This is now the phase where more and more AI is being used in certain applications. It's a slow process, but it's a process of acceptance. So some people are open, some people are critical.
So
Depends.
why I'm saying is that, see, this one is the glucose sensing. So here we have a continuous monitor where few misses might be there,
Yeah.
where you are trying to fill the gap with the AI model and then forecast, which is pretty much robust. In some sensors, it may not be too much. So less of the data, and then you use more of the AI model to do that. So
Yeah.
how the clinicians respond to those things?
So there are also like so it's, I would say that, okay, like for example, if you are there are certain papers and some work, people have actually tried to estimate blood pressure from PPG signal as well. Okay, so the same oximeter that we are using, they try to estimate blood pressure. So some people are acceptable towards that, but they also say that, okay, this is not as good as the cuff monitoring that you have. So it varies. So it also varies on the application. If you want just a qualitative estimate, then okay, it's fine. But if you want a quantitative, very accurate, then you require a good method. So that's.
So Dr. Sanjiv, can you comment on how the clinicians in the UK have the confidence on these type of the models? And
Well, I mean, I would just say it's the same thing what Khalid mentioned. It's well-being versus clinical judgment, right?
Yeah.
And as far as AI is concerned, I mean, I know a lot of oncologists have started using AI picture-based,
Yes, yeah,
clinical
images.
decision support systems, wherein they are more happy to use AI for their clinical decision support system. So, in fact, now we use the term clinical decision support system, wherein you are using the AI tools, and they have a lot of confidence in that because they need more support, and this AI is based on machine learning and things like that. The models that clinicians have started understanding. So some of the latest the new clinicians who have just passed out are now doing a PhD in the area of AI so that they
Yes.
can understand it better
Yes.
and understand the technology better and have more confidence. So
Yeah.
it's all about having confidence in the technology.
Yeah.
And I think with time, more and more clinicians are getting involved in machine learning and AI, and they themselves are getting involved in handling all the records. They're actually going back, digging all the previous records, and try to make sense of the data. So they try to do what we call as the whole process of stratification is based on AI and machine learning. So there's a big effort in this direction, especially by the clinicians in the UK. They're having more confidence on this technology.
Yeah, so I also have a collaborator. He works in the US. In his university, actually, they are actually running some courses specifically for clinicians. So clinicians can actually learn about these AI methods, and then they actually it might help in increasing the acceptability of AI in these things. Yeah.
Any other question, guys?
So I wanted to ask that does it take into account like the relation between two analytes or multiple analytes while there is a missing in data?
Missing data point.
Yeah.
No, the assumption is that that is already dealt with in the sensor itself. Okay, the sensor itself, we are assuming that the reading that we are getting, it's specific to glucose only. So the imputation, the data replacement or synthetic data generation is not considering that. So it's working on the assumption that the sensor is giving me good data.
Okay.
Any other? So I think we can conclude the session.
Thank you. जी हाँ अभी डाउनलोड कर रहे हैं हाँ
I think it's this one. Huh? So welcome back to the next lecture of this GIAN series thing. I think the first lecture that we had this morning was more on how we were combining the receptors together to get a sensitive detection. Now I'm going to basically give this lecture again. This is some unpublished results here. So again, very things that are still ongoing in my research group, and I'm happy to share this here. This whole particular talk, which is going to be on AI-enhanced subdermal wearables for real-time biomarker monitoring in ambulatory livestock. It's good that we have it at this time because we've just had this lecture on machine learning and AI tools from Professor Khalid Mirza. So I think this will be kind of help you understand exactly as to how the AI could also be used for developing wearable devices and getting the tools that you need to be able to predict the right values, especially if you're monitoring in ambulatory livestock. As far as I'm aware, there isn't much work that has been done in animals. Wearable technologies are by themselves, as I say, they are kind of newer technologies that are being used. I haven't seen any evidence, any literature evidence of any molecular, biomarkers being measured using these subdermal technologies, using these wearable technologies in animals. So I'm very happy to present to you something that's unique and something that's new, that's novel. And this is going to be the outline of the talk. I'm just going to tell you a little bit of the historical resume here. I'm going to tell you exactly about why do we need these studies, what we've been doing over the last few years. And I'm going to be telling you a little bit about the need for subdermal wearables in animals, how these subdermal devices can be used for real-time monitoring. And again, we're going to have a look at the AI tools that we have for these subdermal devices, and then look at basically when you do a study in whether it's human or animals, you normally have two parts. The first part is the tolerability studies, and the second part is looking at the performance of the wearable subdermal devices. Okay, since I mentioned the term subdermal, now you've all been here, and I hope everybody is wide awake. What are we referring to here? When I'm using the term subdermal, what are you expecting from the devices? What are you expecting the devices to look like? Well done, micro needles. You're looking at something that's sitting down in your subdermal space, something that's penetrating the stratum corneum layer and going down and sitting nicely in the subdermal space, bathed in the subdermal interstitial fluid. So the talks that we have now and the following talks are going to be related talks. This is more the studies on animals, to show you some real pictures, some live examples, so that you will believe that we can actually achieve a lot using wearable technologies by applying them in a different arena. In this case it would be animal welfare, and then I'll do the final talk, which will be on subdermal wearables, which will be in humans as well. And I'll also give you an example, or I'll show you how we normally implant a continuous glucose monitoring sensor in humans by implanting one in myself, but that will be part of the next talk here. Okay, all right. So we did a presentation a few years back. This is clearly, as you can see, although you have the logo of University of Liverpool here, there was this is the nice buildings that we had at Swansea University. We did a talk on continuous monitoring biosensors for farm side monitoring for endemic and metabolic diseases in animals. This was done long time back. We've presented this in 2023 conference and then in 2025 conference as well. Let's have a look at the need for wearable technologies, right? Okay, why do we need wearable technologies? Everything actually goes back to this big problem of global warming, right? We all know there's this process of global warming going on, and what has happened is we've had multiple United Nations assemblies where people have taken an oath, we're going to cut down on the emissions, cut down on the emission of gases that go and cause the global warming. And people have set up targets for 2030, 2040, 2050. Right? There are different targets. You might have seen one of the ways people are trying to cut down on the emissions is by moving away from burning petrol and gasoline, petrol and diesel, and moving towards more electric cars. I think that's one of the trends that you see. One of the other trends that's happening is in the area of animal husbandry or animal farms. It is believed that keeping animals, especially cows, cattle, it contributes towards global warming as well. In fact, the poor cows have been blamed for 16% of the global of the emissions that take place of the gases that cause the global warming. So what are people doing? I can tell you a little bit about the landscape that we have in the United Kingdom. People are doing what we call as dietary changes in animals. especially in cows. They're trying to modify the diets, giving them more diets so that they produce less and less of methane. And anybody has been to a farm, animal farm? How does it smell? Not very pleasant, right? You can smell a lot of methane in there. It's very, the cow dung and all those things. A lot of emissions there, a lot of methane being emitted there. So that's the main problem. So we need some technologies. I mean, of course, if you change the diet of a cow, it's going to affect the well-being of the cow as well, isn't it? I mean, suddenly, if think as humans, right? Suddenly we all get, I mean, you're all very young, but normally once you hit a certain age, you all still want to maintain, that your metabolism slows down. And then what do you do? And you've been advised to cut down on your carbohydrates, cut down on your fats, and all of us then end up looking into supplements. All of and us, especially people who are vegetarian, they say, okay, fine, I can't have much protein because I'm a vegetarian. So what do? I start looking into substitutes. You'll start looking into soya protein, right? You change your diet. You change your diet, it affects your lifestyle. The energy levels might increase or decrease, right? It affects your well-being there. It affects your mental state there. The same thing is happening with the animals. It's just that as humans, we can express ourselves, the animals cannot, right? So you need some kind of a monitoring system which can monitor the well-being of the cows. Because what's happening, as I said, because they've been blamed 16% of the for emitting those gases, 16% of those gases, you're trying to cut down on the numbers. You're trying to change their lifestyle. So clearly you can see that when you're doing these kind of changes there, when you try to extract maximum out of a cow, you try to increase its milk productivity and stuff like that, you change its diet, it all those changes will bring in some form of metabolic changes, some form of endemic diseases in cows as well. So that's one of the main reasons that we need to have a continuous monitoring system or a continuous monitoring sensor in cows so that we can monitor their well-being. How can you monitor the well-being? By monitoring the levels of glucose. That tells us about the energy levels in a cow. How can you measure the stress? You can measure stress hormones such as cortisol. You can measure lactate in cows. Another thing that you see in cows, especially which you see one of the metabolites during excessive stress is ketones. The common disease in cows is ketoacidosis. It's very common in cows, right? So that's one of the things. The other thing that happens in cows, especially milking cows, is after giving birth to the calves and between lactation, there's a period of time when these cows are very much prone to endemic and metabolic disorders, right? You might have heard about cow lameness. The cows can have diseases like milk fever. The cows can have diseases like mastitis. I don't want to show you those pictures there because those are disturbing pictures, because as I said, in humans you have a problem, you can express yourself, but with the animals you can only see it visibly. But why wait till the end to see a cow reaching a state where it's got lameness? Why not identify that before? As in humans, we try to make every effort to predict the diseases 15 years in advance for Alzheimer's. Why shouldn't we have something similar for cows, which is giving us for the farmers, it's the livelihood. And as I said, we're putting in a lot of stress on these cows, a lot of metabolic stress, a lot of stress that causes endemic disease there. So the reasons that we need wearable technologies for cow or for livestock is because of the metabolic diseases that impact the productivity in transition cows. When I say transition cows that are transitioning between giving birth to calves and also to before they start producing milk, right? That's the period. Those are called as transition cows. In addition to that, you also have cows that are finishing, or they have their own life cycle wherein they are productive. They produce calves, right? And these are very expensive animals. In the UK, we have cultures where you have farmers or you have farms which have got several thousand of cows, and all of them are taken care of in one very close surroundings there. So there you have to look after the well-being. In India, it's very scattered. You would have farmers who would have 2, 3, 4, 5 cows, but I don't think they have this whole idea of putting them all together under one particular roof and try to look after them. So it's a big need there. So in the UK itself, we know that the diseases that in livestock can cost the UK agriculture about 600 million per year, and it can affect the animal welfare and trade as well, right? Anything happens to the animal, you have to put it down, right? Now that's not very good. And you can tackle these by successfully coordinating approaches. You can bring in your high quality science, especially your biosensing technologies, and you can tackle these kind of problems there. And these technology, and you can the problems that the cows have, they impact the farm income. Yeah, if the cow stops producing milk, there's a loss of productivity, there's premature death, and there's what we call as mandatory culling. I mean, nobody wants to see an animal doing nothing there. You can't even use, so what happens is straight away they end up being culled. That's a big problem there. So you can clearly see here from this particular slide, there is a need to bring in the biosensing technologies so that you can monitor the well-being of cows as well. Besides, the other thing is, how do humans keep, I mean, we had a BBC documentary which documented sometimes these cows are badly treated, right? They all go through this milking parlors. They have a robotic system wherein while they are queuing up and being fed, they are being milked as well by the robotic arm, which then basically, it basically tries to extract the milk out of the udder and stuff like that. So it's a very systematic way of doing things, but sometimes it's not an ideal case scenario. Sometimes, you may have some funny workers there, some people who may not be in their best mental health, and they may not look after the cows very well, right? What does that affect? That affects the final productivity, that affects the quality of the milk, right? More stress, more hormones in the cow would lead to milk that is contaminated with those hormones. As I said, in India, you have the problem of farmers using oxytocin to increase the milk productivity, right? And oxytocin in milk being consumed by humans is not the best thing. So you need biosensing devices which can do point of care sensing to tell exactly if they're using this or not, they're using the oxytocin, high levels of oxytocin or not. It's like using fertilizers, right? Fertilizers are good, they increase the productivity of plants, but when you exceed a certain level, it can start destroying your fields. It can start destroying your long-term productivity as well. Same thing happens with cows as well. Too much oxytocin starts killing the cow itself, and it's also not good for the humans consuming the cow milk as well. Okay, so let's move to the next slide. So if you go to a farm, I mean, this is a picture of a UK farm. This is one of the farms from Wales, one of the Welsh farms. You do see devices there. It's not as if technology is completely new when it comes to animal healthcare. You see, these are some of those devices that have been inserted in the animals just to see the effect. These devices are called as intraruminal devices, quite very invasive. If you look at they are actually making contact with the alimentary canal, the rumen of these animals here. So you can see they've been they're very invasive devices, and they've been actually, the term would be implanted because they go quite deep into your body. They've been implanted in the animals here. You can see that those in the example of cows and in sheep as well. So as I said, biosensing technologies, I don't think it's a new thing when it comes to farm. I have seen people using biosensing technologies. This looks very similar to a glucometer, doesn't it? Right? This is where you have a strip which has got your biosensors, your bioreceptors on it, and you can see there's a drop of blood here, and you're getting a reading for the ketone here, which happens to be 1.3, which happens to be a good reading, right? That's all fine, but what's the problem here? The problem is, look here. How are you getting the blood out of this thing? Can you get capillary blood out of animals? No. Why? Because of their paws, right? Because they have dead, it's very hard to extract capillary blood out of their feet, right? So the only way you can sample the blood is by extracting the blood from the tail, right? Again, poor animals, they can't express themselves. We do not know whether they are in pain or they're not in pain, right? You can't do a visual assessment method here and ask the cow to answer you some questions, right? So this is where you just look at the cow. If it moves around a little bit, if there's a bit of discomfort there, that's your sign that the cow is in there. So what I'm trying to say here is that, if humans need wearable devices, wearable technologies to address their healthcare needs, their well-being issues, animals need it more, because unfortunately, you can't do this tolerability assessment with them wherein they can express themselves and say, you can only see it in the form of signs. And sometimes those signs are very late. When you suddenly see a huge tumor growing in there, and you're thinking the cow is not, how did this suddenly appear? So they need early diagnostic systems even much more than humans, because they do not tell us anything about their symptoms. All right. Okay, so these are pictures that were taken by myself in the different farms. This is the picture of this. This is the cow, but this is the gentleman here is one of my collaborators, Professor Dr. Joe Neary, who is an animal scientist, is a veterinary scientist, one of very enthusiastic collaborators that I have, and I think I owe most of my animal studies to him, because he has taken interest in this whole thing and actually given me all the tours of the farm. All right, now we spoke a lot about how do you start working in animals, in humans, right? So to do clinical studies, what do you need? Before you do clinical studies, of course you need a clinical collaborator. What is the other thing that you need? Ethical approvals. In the case of animals, the ethical approvals come from the Home Office, right? And it takes a long time to get ethical approvals. So we had a project which was a mix of AI and biosensors, but used for animals. So we had a project which was funded by the Welsh government. It was called the Welsh Digital Nation Accelerator Program, wherein we were trying to combine AI to sensors, because we knew that continuous monitoring would give us a lot of data. You've already seen that from the last talk. You saw that predicted data and time lags and stuff like that. We've already had that talk there, so I'm not going to talk much on the AI side of things. We were very keen on starting the studies. The only way we could study or we could use our sensors was in freshly dead animals, right? So I went to this farm. This is Joe Neary here, who's hidden in the picture here. That's myself back, I think this is about 2017. No, this is about 2021-ish kind of time. These are my students, Georgetta and I think this is Zenny. Both of them were my students there, master's student doing the research project. And as you can see here, this is a freshly dead cow, not a very clean environment. I've cut off the picture images here, but you can see there are blood there. So what happens is in the UK, it's a practice that when you have a dead animal, you bring it to a place before they are slaughtered, or they're already dead. Before they are kind of disposed of, or before they are incinerated, you bring them to a place. You open up their head, they open up the skull, take out the brain, and you look for any signs of any nodes that could say that there's mad cow's disease. Mad cow's disease is very prevalent in the UK, but because the symptoms are not very evident, that's how they do a physical examination, right? You're absolutely right. It's a post-mortem examination, right? Wherein you open up the skull, take the brain out. So while we were sitting and we were working on this freshly dead cow, which I can tell you from my own personal experience because I felt it as well. It was very warm, in terms of the body temperature. There were other cows brought around, and so I had two students again from India who couldn't stand that scene, who couldn't stand the stench that was there in the farm. And so one of them had to be taken off the farm or out of this facility to another place so that he could breathe, and otherwise we were worried that he might throw up, right? So this is just to show you that these are the conditions under which we had to do the experiments there. So again, you can see that what we were trying to do here is we were trying to see which is the best part of the animal, which is the best anatomical location in animal which would give us a good signal there, right? We were trying to look for chemical signatures. As I said, you're pushing the needles through the skin into the interstitial fluid, and then what happens is in your interstitial fluid you will, again, it's a complex mixture. If not anything, you will have glucose. Even if that glucose concentration is dying over time, even if it's kind of, there's a exponential decay there, you would be able to measure something there, and the current values would be typically between microamps to nanoamps kind of range. That's the range you're looking for. So what we did was we managed to shave off the neck. This here underneath the adhesive, the plaster is the microneedle device. Again, at that time we were using quite large potentiostats. This is our CHI potentiostat that is sitting in a box there, and that's a laptop that we have there. Again, farm conditions, right, makes you think. And the reason I'm showing you this slide is just to give you an idea. It's very cold. If I left my my laptop there running overnight, next day I come down, it's shut down completely. My potentiostat will not work in farm conditions. It's so cold there. The temperature could drop down to as low as -5, -4 degrees, right? And at that temperature, none of these, no matter however good, whether it's an EVM or a PalmSense potentiostat, I think all of them would fail. So just these are all food for thought for you, so that if one of you decides to go into using these wearable sensors for animals or developing these, what do you need to keep in mind when you're designing these products there? All right. So this is chronoamperometric measurements using two different sensors, showing you're getting nice stable currents here, which are an indicative that you can have the sensors in the neck area of the cow as well. Why the neck area? Skin cleaner, blood supply, good blood supply there as well. What else? What do cows have around their neck? Collars, right? I mean, that's a standard practice everywhere, right? Even in India, cows have collars, right? And then what do they have on their ears? They have tags. Do you think ears would be a better place? I mean, they have tags, right, which are like proper plastic tags. Do you think you can have devices there because they're used to that? I mean, those are the ones where you proper piercing there, right? Do you think we can put the subdermal patches there? Yeah. What do they do with the ears all the time? What do you think that will do? Artifacts, motion artifacts. Okay, just food for thought, right? Okay, so these were some experiments some preliminary experiments that were being done just to kind of get some idea about what is going to be the best anatomical locations, right? Which one we selected, I'll come to that later on. I'm just telling you this in the form of a story just to tell you how this whole work kind of nicely evolved and has got to the stage where we are today, where we are probably the first group to be able to do apply wearable technologies, especially subdermal wearable technologies in animals. Okay. In the meantime, I also mentioned to you about this project. I mean, this slide will come up again and again. We had this DASA project wherein we were working on subdermal sensors, which were for simultaneous glucose and lactate monitoring. We'll cover most of this in the next presentation there. And here we were using our platinum subdermal sensors, and we were getting some data coming out of it. I think I've already shown you how the data was looking like, right? The data that was coming out of this was looking very, very, the raw data didn't look very good. So what we had to do is we had to bring in the AI tools. I'm not going to talk much about that because we've already had a lecture on that, and we were making sense of the data here. So it clearly made sense for us that if we had to use AI tools for humans in ambulatory conditions, again, we've spoken about this whole thing of doing studies in steady state and ambulatory conditions. Ambulatory conditions is a different world, right? Any small movement will give you this. So the animals that we are looking into are going to be in ambulatory condition, right? You're not expecting a cow to be sitting there in a clinical research facility lying down on a bed and you're giving it tea and coffee and having a potentiostat there and very much relaxed. None of that is going to happen there. This is cow, harsh conditions, different, really low temperature. And again, if you look at the cow environment, I mean the farm environment, it's full of feces everywhere, it's full of cow dung everywhere, and the cow themselves are actually, if you look at different body parts of the cow, they are almost covered till half of their body is covered in cow dung. And it's not like like that they're getting regular showers by humans or they themselves are having showers. Maybe in India they have the opportunity to go and have a shower, a bath by themselves, go to the ponds and enjoy themselves a bit of a dip there. That doesn't happen in the UK farms. So the reason I mention this is not to make you laugh. The reason I mention these are the practicalities. This is the real world scenario, right? If you think, for example, I was very interested in the udder part of the cow. Why? Not because I was interested in metabolites. I'm interested in more biomarkers of diseases. And the udder, as you all know, if we clean the udder, you can see it's very pink in color. Why is it pink in color? Because it's highly vascularized, right? And when something is highly vascularized, that means that there's a much better equilibrium of the subvascular plexus and the skin interstitial fluid. That means that you would expect to have less time lag between your target biomarker and in the blood and in the skin interstitial fluid. That's why I was very interested. But when I looked at the farm side environment and when I saw the udder is almost half of it is covered with the feces, I said, oh, this is not where the sensor can go, because your sensors, again, these are molecular sensors. They are very sensitive to any chemical, and your feces is full of different chemicals. So you're definitely going to have cross contamination. Okay, the idea of this slide is to show you that, we had identified through the human studies that we have AI models, right? And these AI models can help us in predicting much more accurate values, can help us predicting or minimize, in predicting the decline in the performance of the sensors. It can help us in looking into things like artifacts and stuff like motion artifacts, movement artifacts, etc., etc. So, and these are the results that we got, which made a lot more sense when we brought in the AI side of things. So we said, okay, if you bring in the AI side of things, 3-day monitoring, the data looks very good, clock grid error looks very good. We'll go into these in more detail in the next lecture that we will have before the lunch. But just to show you a glimpse of what the message here is, AI actually helps in improving the quality of your data. So coming back to the animal project here. So what's happening here? You have your subdermal devices, right? You want to apply your subdermal devices to monitor the well-being of animals, especially in farm side for cattle, in livestock monitoring. The problem you have, the challenges you have is, number one, thicker skin, right? The animal skin is much thicker. So clearly what you need here is, you need long microneedles, slightly longer than what you used in humans. And you also need, ideally you also would need an applicator, and you would also ideally you would want to work on the animals without having to shave off their hair. I mean, cows is fine. I mean, the farmers wouldn't mind seeing cows that have been shaved off a little bit. But if you try to do that with the horses, horse owners take a lot of pride in their animals, right? They don't want you to bring in a clipper anywhere close to the horses, right? You take a clipper and say, can I shave off this part? They said no, right? So we are lucky, we still have access to animals, to cows, and we are allowed to shave them off. And as I mentioned, I gave you that interesting story wherein I found heart shapes drawn on the with the clippers done on the cows, and I later on realized that I'd actually sent my students to do these studies on a Valentine's Day, and that's where I could see all those heart shaped signs there. But anyway, that's a different story. The other thing that happens in animals is they have a complete different metabolite range. In humans, the healthy range is 4 to 7, and cows have got a much lower baseline glucose. They've got different lactate thresholds, right? Which means that you need to be, your dynamic range isn't 0 to 20 millimolars. You're actually looking at something which is between 0 to 10 millimolars. That's the kind of dynamic range you could look at. And again, with diabetics, with humans, when you have diabetes, the symptoms are very much visible, and you can identify it. When you see somebody's feeling thirsty, has this, is going to the toilet quite often, there are some symptoms that can be picked up in humans, and based on the symptoms, it's easy to predict. Animals, how do you predict whether a cow is diabetic or not? You definitely need the sensors there, all right? So they have a different metabolite range. Reference sampling. Again, reference sampling is very hard. I showed you that picture there, right, of the cows being sampled. Look here. This is how reference sampling is done in animals, right? Because you cannot get any capillary blood, right? Can you get tear fluids? It's very hard. You can't get regular. Can you get sweat out of these animals? Epidermal sensors? Anybody thinking of that? Yeah, wrapping around a whole, the size of the animal doesn't allow you, and they don't have fingers. So how do you make small devices that can be wrapped around the fingers? Yeah, you will have to wrap something around the whole body to collect sweat, right? Even if you want to measure urea, which happens to be the best biomarker for epidermal sensors, I don't think you can so there's no case here for epidermal biosensors, right? No case here for epidermal wearables here. Okay, so coming back to the challenges, and then tolerability assessment. I mean, any study that you do on validation of devices, any study that you do on your medical devices has two main parameters. One is, how well is it tolerated? Is it going to be accepted by the user? Number one, how well is it tolerated? And the second thing is your performance. Performance is an easy thing, that's fine. Again, performance, there is a challenge in performance, I'll come to that as well. Performance will come from the differences, problem with the referencing thing. So I think the problem we have here is with tolerability is that you cannot do a visual assessment method. So I'll show you the next presentation on human studies, wherein I'll show you how we were doing a visual method. I think I might have already shown you some slides wherein you insert a cannula, right? And then you basically take the cannula and you compare against the cannula. Right? So here you cannot do that kind of thing. You will have to, and you cannot keep on doing biopsy of the animal skin and looking at the biopsy to see if there is any inflammation or not. You have to do visual assessment here, but that's not going to be, that's a visual assessment that the human has to do rather than the cow telling you what the pain scores are looking like. Okay, all right. And then finally, as I said, bioinstrumentation constraints, right? The farm side, to have devices that work on farm side, they have to be rugged, they have to be waterproof, and they have to be lightweight, especially if you're looking at something that has to be carried by the cow, especially if it's a wearable device where you're looking for monitoring continuously over a longer period of time. If in humans we are monitoring over 14 days, in animals you need it for even longer periods of time, right? So those are the main challenges that you have when you are trying to apply subdermal wearable devices in farms. Okay, so what did we do? This is our design. As we said, I mentioned the first thing you need is you need sturdier, you need much more better you need micro needles. So obviously we were using polycarbonate ones. The polycarbonate ones are not ideal for animals, because as I said, sometimes you may be in a scenario where the farmer might refuse to shave the cow, right? He says, no, I'm not going to let you use your clippers. So you need much more sturdier needles, right? So which are the best needles that you can use here? Stainless steel, right? What is the best source of stainless steel needles? Lancet. What else? Lancets are CE approved, so I think Lancet is a good choice. What's the height of a Lancet needle? Have you ever tried? 3 centimeters? 2 centimeters? What else can you use? I'm glad you're doing this. At least you're getting an idea what the measures, because the thing is, if you don't visualize what I'm talking about, it defeats the whole purpose. The ideal height would be something around 1 millimeter, right? And what can you use for that, which is commercially available and CE marked and very cheap to procure as well on Amazon? Acupuncture needles, best thing to do, right? You can take them. What's the problem with acupuncture needle? Sorry? No. Robustness, from point of view of robustness, perfectly fine. I mean, they're deliberately made thin so that they can do acupuncture without causing any pain. But even, has anybody undergone acupuncture in this room? Even for fun? No? You should try it, right? When they put the needle in, sometimes when they push it back, they put the needle in, they insert the needle, and then there's a broad thing, then they push it in with the thumb or something like that, and then when it touches your nerves, you can feel it. You can get a tingling sensation. Did you say you didn't try an acupuncture needle? Okay, you've read it. Yeah, so you can use acupuncture needles. So these are pictures of acupuncture needles. I've got much more clear pictures which will come forward. And then again, you're looking at standardization of the insertion, right? The term I use with subdermal devices is insertion, not implantation, right? What is implantation? I'll show you when I show you the CGM device. This is all insertion. Now, if I gave all of you, a subdermal, a blank subdermal bare structure, if I gave it to all of you and asked you to push it into your skin, everybody would use different forces, right? But that's not, when you think about the final product, what you need something which is a bit more standardized, right? You need something that gives you the desired force of 10 newtons. So in this particular case, we designed an applicator, and also for the instrumentation, as I said, the instrumentation doesn't work. You need the instrumentation to be nicely cased and everything else. We designed our own instrumentation. We've got a PhD student who's a very talented one. I wouldn't say master of all trades, but still jack of all trades. But he can design his own potentiostats. And I think these are based on similar, if you remember the module we had, the lecture we had yesterday from Professor Khalid, wherein he said you can design your own, you have those existing designs on which you can build your potentiostats, whether it's single channel or multiple channels. I think he's used something similar to that, and again you can see from this thing here, he's got one counter electrode, and then you've got your four working electrodes 1 to 4, and you've got a reference electrode here as well, right? Begs the point, wherein basically this thing is actually connected to multiple. So if you look at the design, it's a radial design. So if you look at the needles, you would have, look at this design here, this is more like a radial design. So you have an inner circle, then you have an outer circle, and then you have a further outer circle. So if you have the middle circle, you have one, which is your reference electrode, and then outside circle you have your working electrodes. So you can have 1, 2, 3 and 4, and as I mentioned to you, as a thumb rule, your counter electrode should be either equal to the size of your working electrode or twice the size. So what you can have is, you can have an outermost circle where you will have your counter electrode would be 1, 2, 3, 4, 5, 6, 7 and 8. So you have a multi-channel, meaning 4 channels, to allow you to do 4 of these, to measure 4 different analytes, or same analyte by 4 different channels. Okay. And then, as I said, these are your working electrodes, and this is your reference electrode. The outer ring is your working electrode, and the outermost ring is your counter electrode. That's what you're seeing in this particular slide here. Yeah, is the design clear to everybody? Can we move to the next slide? Yeah, everybody happy with it? Okay, all right. So, oh my God, what happened here? The font is completely changed. Apologies for that. This is just to give you a little bit more information on the applicator, because I think we had some questions on the applicator on day one, wherein somebody asked me what are the conditions for the applicator. I mean, it's standard rules of physics that you apply here. F equals kx, which is basically a force exerted is directly proportional to the spring displacement, and the constant is given as the spring constant here. So what we have in the applicator, it's basically a spring loaded design. If anybody wants to look at the exploded view, I can give you that, because I think just last evening I received the manuscript on that particular design. So if you need the thing, I can show you that as well, right? But what you basically, the main criteria or the main parameters that you need for are, you need to apply a force of 10 newtons. 10 newtons is not too much force. A moderate thumb pressure is 10 newtons. So if you take a coin and put it on your skin and apply moderate thumb pressure, you can feel that. But that's you won't feel that much because you're applying 10 newtons through the needles. So clearly the force is the same, but the pressure changes because you're applying it through nearly 20 micron fine tips. So you can imagine the pressure goes up quite a lot. But the important thing here is, if you should hear a click. So you have, it's a spring loaded device. It has to be inserted at a certain speed, and that speed is 10 millimeters per second. If you go anything less than that, you can cause damage to the skin. If you go any faster than that, that can also cause damage to the skin. So you need an applicator that gives you 10 newtons at a speed of 10 millimeters per second, right? That's the example of your applicator, or the conditions for your applicator here. We did some insertion studies. I mean, as I said, it's very important that you see what happens with the insertion studies, and we tried to use the CGM sensors as the secondary reference here. We were doing venous blood, which we were mainly looking at the tail blood, but we also had the CGM sensors. The CGM sensors basically have got a wire, right? And these wires are normally pushed through a hollow microneedle into the skin, into the subcutaneous tissue, and then, so this is just to show you when this is pulled out, for some reason it's lost its shape. It should have been straight, but it's lost its shape and it's gone all the way bent. You can see that the CGM sensors that are designed for humans are clearly not very good for animal studies, right? That's what you're seeing here from the CGM sensors as well. So clearly the data that we get here is not very much reliable. This is picture from one of the CGM sensors we have. I need to see more of these pictures to kind of confirm that all the sensors were failing in animal conditions, okay? And here are some this thing. So this is some mechanical property testing. You're basically looking at the mechanical properties of your acupuncture needles. These are your nice, acupuncture needles that you can see in height, about 1 millimeter in height, right? The scale bar, if you put the scale bar together, these are 1 millimeter in height. So these are pre-insertion. So we take a picture of these before the insertion, right? And this is post-insertion. So you push it in and then take it out. And this is, I think, post-insertion. This is after nearly 28 hours of insertion. So you can see here, I mean, they look very mechanically robust. You have some debris here which comes from the whatever is there in the skin interstitial fluid. You can always have some salt layer formation around the sensor surface. There can also be some biofouling effects there as well. So this could be one of the combined effect of all the biofouling that takes place and all the debris that comes out when you're pulling them out of the skin here. But the message that I'm trying to give you here from this slide is the difference in between the pre-insertion pictures and the post-insertion pictures there isn't much difference there. Can you see any mechanical failure there? Anybody? It has slanted. Okay, I think it's not. It looks slanted because it's not a comparison of the same like-to-like image. So if you look here, the reservoir. This is another needle here. This is so it's taken at a different angle. If you look at this reservoir, this thing on the PCB, it's at a different angle. But I mean, when we looked at the images there, we didn't see any mechanical. Any failure of the mechanical properties. So it looks like it's sturdy enough, it's robust enough to be sitting in there for 28 hours. That's what it looks like. Okay, and then basically this is basically how the cow was dressed. As I said, we try to apply the sensor underneath the neck. You can see everything is going in there, including the instrumentation as well. Everything is nicely, and these are the wires there. which have been nicely, the cow has been nicely dressed on the Valentine's Day to be ready for those measurements that we need to have. And this here is basically a video just to show you how the insertion process is done. That's your applicator, spring loaded. You see they've shaved off the hair there, and now they go in there with the applicator, hold it in place, and then they push the piston. Here we go. So you can see that you might have seen the hair of my student. I think there's no difference between his hair and the cow hair. Both of them have got quite long hair there. And here's the device that has been inserted, not implanted, right? Inserted on the skin of the animal. Ideally, we would like to have it even if without having to remove the hair. That's the kind of thing that we would like to have. Okay. Sorry, excuse me. All right. So that's the kind of showing you how the sensors are in action now. So you can see there are 3 wires coming out of it. This is just a single channel one, right? Clearly the white one would be reference, the red one would be working electrode, and the green one would be counter electrode, or something like that, depending on what the labeling, convention they're following. Okay, let's move to the next slide. Okay, the next slide is on the tolerability studies. It's very important. It's very, very important that we do a study of tolerability. How can you do tolerability studies in animals? You can't do a visual assessment, right? The cow isn't going to respond. What's the way you can measure tolerability? Anybody here? Yeah, you look at the skin. Look, after you've removed the devices, what happens to the skin? How can you measure tolerability during the insertion process? If it moves, if it does what it normally does, if it keeps on grazing on grass, okay, fine. That means that it's all. If it performs its normal regular functions, that's the best way to say that, right? And I think I might have a video to show you if it's performing its normal functions or not. Okay. Now here are 2 different. So these are 2 dots. Two circles that you can see here. The yellow circle is what happens when we remove the subdermal device, and the red circle is the one where you remove the Freestyle Libra device. Now, again, I say Freestyle Libra, and we've spoken a lot about iontophoresis, and we've spoken about the gluco watch thing here. What you see here is, I mean, it's probably not very clear because there's too much light on the screen here, but on my screen I can see it very clearly. You can see if you did a high resolution image there, you can see there's some red marks there, and these red marks are a sign of inflammation, right? These red marks are a sign of inflammation. And the subdermal variables, can you see anything there? There's nothing happening there. So that's the first sign, and as I said, it's not just one image. You have to analyze multiple images to be able to ascertain, because even if humans are different, clearly cows are going to be different as well. And in cows, the other problem you have is they all come in different colors as well, right? Some of them are black and white, some of them are red and white, some of them are brown and white, so they come in different colors as well. So clearly there's some difference in coloration as well, and sometimes it may be the inflammation might be clear, sometimes it may not be very clear. So we have to analyze multiple images to be able to come to this particular conclusion here. Okay, so this is, I mean, good enough evidence. Would you say good enough evidence to say that if you have multiple images to show these kind of images, good enough evidence to show that they are tolerating the subdermal wearable devices? Although they did not complain about the Freestyle Libra as well. Although when we had the Freestyle Libra and when we used the applicator, you could see that they were twinging a little bit, in the sense that they would just have, you could see a sudden movement there, but there wasn't any complaint after once the devices were, it wasn't as if that they started, making a lot of noise or anything like that. Okay, this is a video to show. So we kept the cow a bit into the enclosure because sometimes when they go for gazing, we didn't want it to kind of take the sensor and disappear and come back after 14 days. So because we know our bioinstrumentation did not have that battery life there, so we kind of kept it in an enclosure. Also just to keep an eye as well, if there are any side effects kind of thing as well. And we had the animal scientist there just to show you nicely, so doing what it normally does, right? And all the sensor and everything is nicely covered goes under the collar side of things as well, right? Okay, so that's the tolerability studies. You can see it's quite a cold day, and you can see all the methane coming, it's blowing all the methane through its nostrils. Now let's have a look at the performance. What happens in terms of performance? Okay, now what we see here is the data. I mean, this is how the raw data actually looks like, very noisy. I mean, this is actually, and I'm showing you the raw data. I'm not making it up at all. And then you use your smoothing your when you're plotting, you can use your smoothing functions, and then you can smooth the data. So all this data is nicely smooth to kind of give you these kind of peaks, right? And as I mentioned, we take capillary blood at times. So this is a capillary blood which happens to be at a time when it was already peaking up, and then we were following what's happening there as well. Now again, it's not very easy to take tail blood from the animal, and sometimes even when you take the tail blood from the animal, it doesn't give you a good reading. Sometimes you get error response, and that's a waste of that sampling that has been done there. So we did multiple things. I'm just showing you some of the studies. Like this one has been done over 26 hours. We had the CGM sensor that was implanted here. It worked, and then it suddenly went quiet. So there's this huge gap here between this region here. There's no data on the CGM sensor, and then it suddenly woke up and started working up again. And you can see that, you can see those peaks which are going up and down, which actually didn't make any sense. Would I be worried about that data? Would I be worried about the correlation of that data to my data? No, I wouldn't. Why? Because we've already shown you pictures where the CGM data, the CGM is actually failing mechanically. If the sensor is breaking down, this data doesn't actually make any sense. All right. Okay. What will make sense is these measurements from this capillary blood glucose, or the vein. It's not capillary blood. There's no capillary blood here. It's the tail blood that we are normally looking into. That'll make more sense here. So we did multiple of these. This is just to show you. This is to show you measurement over the entire day, how the profile actually looks like, and then how we process the data here. Again, this is a biaxial plot wherein the sensor is actually measuring the current. So the current values that you get here, current and nanoamps, this is the sensor measurement, and you're trying to correlate that to the blood glucose measurements that you're doing by obtaining samples of the tail blood. Okay, and if you zoom into this, we also add in the time at which we've given the boluses. I don't know why these are not missing. So when you zoom into this part, you can see that when the bolus is given, bolus is something when you eat something, you expect to get a peak which is normally referred to as a postprandial peak, right? So PP or postprandial. Yeah, this is a postprandial peak that you see here, and you can see here there's a good correlation of the blood measurement, and after this it rises. You can see that the blood glucose also rises, and then it goes down, and then again there's a small peak here. It depends on what is the bolus that you're giving. It depends on whether it's a low glycemic index. Do you understand GI? It's glycemic index, right? So for example, when you eat brown bread, why do you eat brown bread? Because it's low glycemic index. Why is it better than white bread? Because white bread would give you a much higher peak. Brown bread is more it gives you a broader peak, and it's distributed evenly, so you don't feel hungry over a longer period of time. It's a slow release thing. So rather than getting a huge peak with white bread, you would get a small peak but goes over a longer period of time with brown bread. That's how it normally works. So anything that's giving you low peaks for the same amount of the food, that's called as low glycemic index. Anything that gives you a higher peak, for example, orange juice will give you a very sharp spike. That's a high glycemic index drink. Yeah. Similarly, your energy drinks, your Lucozade and all your Gatorade and all those energy drinks that you have, those give you spikes. Those are not meant to be consumed by healthy volunteers. Those are meant to be consumed by people who are suffering from hypoglycemia to bring them back to normal glycemic levels. That's what they are meant for. But they're being used as energy drinks for sports people. Anyway, that's fine. That's mainly because of the electrolyte balance that they offer you. So what I'm trying to show you from this particular slide is how you can have these peaks. You're seeing the postprandial peaks in response to the bolus 1, and this is bolus number 2. You can see there's a peak there as well. So that's how what happens when you see a zoomed image there as well. Okay, and then when you take all of this data and run it through the AI tools that we have, and then you plot it against blood glucose values, you can see that your points are nicely scattered, with most of your points in zones A and B. 100% of the points are in zones A and B. I would like it to be 95% in zone A, but I would even accept 92.42% given the fact that it's a initial, these are early days for us. We still have to optimize things, we still have to improve things, we still have to optimize our instrument, our equipment, we still have to optimize our sensors. I mean, the sensors are perfectly fine, but there are things around it, the coatings and stuff like that, to make it more friendly to the environment. And I think there's also, all the data will also be used for training the machine learning program, and then we will have a much better AI tool there once we have all of that trained in a nice way. And I think Professor Khalid here will tell you that there's a certain number of measurements you need to do before to train your data, and there's a formula for that, which I think keep on hearing from Sudhansh. I need to do this many readings to be able to then train my model and then use that for my AI tools. So that these are things, and it's nice for you to know that. You don't have to know exactly what the machine learning tools are or what the AI models are, but it's always nice to know that you have these programs which basically help you in cleaning up your data and making more sense of your data and getting more accurate predicted values. The AI tools can also help you in getting rid of false positives and false negatives as well. Okay, so this probably is taking me almost towards the last slide that I have, which is on conclusions. The key finding is we saw a very strong biosensor performance. Does everybody agree with me on that? Very strong performance. Your Clark grid error or Clark error grid analysis shows that most of your points, in fact all of the points, are in zones A and B, which is perfectly fine. It seems to be even better than what Professor Khalid showed you. Some of his points were in zone C and D, but this one seems, although they were simulated data, this one looks to be much better with the experimental side of things. It also indicates very high clinical accuracy for glucose measurements. The instrumentation seems to be very robust. I mean, you have everything on the cow around the collar term, yeah, and you've been able to measure for 28 hours. That's really, really good. I mean, we couldn't do 28-hour studies in humans, continuous, and here we've done 28 hours in animals, and everything is working. There was no breakage, there was no, we didn't lose any, the sensors were performing very well. Yeah, the performance, I expect them to dip down, but the sensors were performing very well. The devices remained stable and function in farm side conditions, and they demonstrated good durability and wireless data transmission. And of course, the use of AI assisted artifact removal. The machine learning pipeline effectively filters the motion. As I said, the cow was not, I mean, it was there, but it was released in the evening. It was allowed to go to the farm and stay there overnight, have a good evening with all its mates there on the farm, and have a good time there. So we are able to effectively filter all the motion and noise artifacts that you would normally expect from those kind of socializing phenomena that was going on there. It also enables real-time signal interpretation as well. That looks very good. And the future outlook would be to have now, we've done glucose. What else would you like to measure in cows? Well done, ketones. You've hit it bang on. Ketones is the next biomarker that you would like to. What else? You'd like to monitor their estrogen cycle as well. The hormones, the fertility hormones, very, very important, because cows have got a very limited life cycle, fertility life cycle, and you have to, farmers try to make the most of it. All right. So
what about cortisols?
Cortisols as well. Cortisols would be the stress hormones that you would like to look at. But in cows, especially milking cows, ketones are normally considered to be much more. As I said, if there's a choice between metabolites and stress hormones, I would definitely go for metabolites, because there's the correlation of these metabolites and the equilibration and the lag time would be much better. The larger the molecule, the longer it would take to diffuse into the skin interstitial fluid. So I mean, Absolutely right. I'm not saying it's a wrong answer. It's a very good answer. But if I had a choice to monitor the stress in animals, in cows, it would be ketones. And that's normally done using beta hydroxybutyrate, which is another dehydrogenase enzyme that's normally available for that. I think that's the conclusions. But I'm presenting the results, but most of these results have been done by my colleagues and my students. This is Sudhansh, who, as I said, a very enthusiastic PhD student. Anu, I can promise you she did not go in this dress to the farm because nobody goes to the farm dressed in red. Goy, also very enthusiastic researcher running around with Anu and Sudhansh behind the cows. And of course, my experienced postdoc Arjun. And of course, this work would be completely impossible without Dr. Joe Neary, who is the equivalent of a clinician here, who is a veterinary scientist and a veterinary doctor. Right, and then of course I have to thank my department for all the facilities they've given me. The work was done at a different campus at the University of Liverpool. We have another campus called as Learhurst campus. The work was done at Learhurst campus, and some of this, as I said, we have a very enthusiastic animal scientist. He also, he's a farmer himself, so he has his own farm, and he allowed us to do long-term measurements on his farm as well. Right, that's it. I think that's the last slide, and I'm happy to take any questions that you may have.
So we have like 4 or 5 minutes more before we start the next session. So any questions?
Give her the microphone, otherwise it won't be recorded.
The tip size of the microneedle array.
I will be very honest here. We didn't worry too much about the tip size of the microneedle. Why? Because these are acupuncture needles, right? In fact, now we always used to use the term subdermal microneedle arrays. These are so we would always call them MN. We call these ones as APN, acupuncture needles, right? Acupuncture needles are commercially available, CE marked, very safe. They form, they have been part of the traditional acupuncture therapy that's normally done. I think it's a Chinese tradition that has been introduced in the world, but it's used a lot in people in the field of sports medicine. I mean, they say that it helps in opening up the cavities and releasing all the fluids and stuff like that. And releasing all the lactate as well. It helps in flow of the electrons and flow of the electrolytes and stuff like that. So it's all known. So we didn't worry too much because these are, as I said, safe, commercially available acupuncture needles. I'm sure if you looked into the tip dimension, you're right. We should probably do a proper characterization, but I'm sure the tip diameter would be definitely around 20 microns or even lower than that.
If you don't shave the cow,
yeah,
then you need to increase the tip size, right?
No, you don't have to increase the tip size. You have to increase the length.
I mean, acupuncture needle won't work.
No, the acupuncture needles that we normally get, they're about this much. If you can imagine, I mean, nobody is going to take a one, because it's very hard to handle a 1 centimeter needle. Oh, sorry, no, we're not talking 1 centimeter. We're talking about 1 millimeter needles, right? It's very hard to hold a 1 millimeter needle. It's like if you take a ruler, it's very small. So normally they come in the form of like a pen. So they have a much wider, towards the end they're much wider, because then it's easy to handle. The front part of it, I think about 5 millimeters is the part where, so we normally take it and we cut it off. We can use the other parts for in vitro modeling and for in vitro studies. The front part we use it only for the animal studies, because we don't, the rest of the part is very blunt. So please don't use that one for animal studies. For animal studies, just use the front part. And if you can find a way of sharpening it and using the entire needle, because there's a lot of waste that's being generated, a lot of stainless steel weight that's being generated, right? The other thing I want to mention is acupuncture needles, stainless steel. It comes with its own set. I forgot to mention this when I was talking about the studies here. It comes with its own set of problems, because what you're trying to do is you're trying to sputter gold or platinum, and then again you're trying to coat, you're trying to electropolymerize a phenol solution or a pyrrole solution which has got the glucose oxidase enzyme on it. But that electroplating step, I can tell you that we had a big gap of about 2 months, 2 to 3 months, where Goy and Sudhansh and Anu had a hard time getting the best protocol for acupuncture needles, because what would happen is if you had different sources, they would have different qualities of stainless steel, and for each of them you needed a different metallization protocol. That was a major challenge there as well, right? And that's why it's always best to zero on and say I'm going to get only one source. We were getting these acupuncture needles from China and from Amazon. So while we optimized on the China the ones we got there, they were very cheap. So it was like, oh, we can get more of these. But then we optimized on that didn't work on the Amazon one. So it was a trade-off. So it's always good to zero in or make sure that you only have one supplier and get a standard operating procedure just for that particular supply of acupuncture needles. Yes, Satya. Okay.
So, sir, there is a sign which sweat on the nose of the cattles. It's sign for the healthy livestock. So the sweat is always present on the healthy animal. So how we can use sweat as a sample? Why we can't use it?
No, you can, because I've seen rings around the cow nose of the cow, right? So if you can design something on that ring which is like an epidermal wearable device.
The upper side of the nose, which is having almost every time there is sweat there. So
Yes, but the challenge there is the volume of the sweat. Is it uniform or is it variable? Because you see they also breathe through their nose, and when they breathe out, I showed you that picture, that video there of the cow breathing out. You could see a lot of, you actually have come up with a very good idea there, but I'm not sure because the nose is very sensitive. Have you ever tried to tap on the nose of a cow without being kicked?
yes, sir, we have.
Oh, I'm sure you. But
doesn't
that's because it's your cow, right? But anybody else's cow?
No, not actually.
So I think the nose is very sensitive. From what I understand, you're the person who owns cows, so you know better than me. But my understanding of cows is, we don't dare touch the nose of the animals in the UK. You've seen how big they are, right? If they just give you a nice little nudge, it's good enough to knock you off for the whole day.
The
So,
major advantage I want to say that cow having the sweat over the time, like 24/7, in healthy cows the
Yeah.
sweat is present there.
Fantastic. That's a good opportunity for you to then develop epidermal wearable sensors. Yeah. But then again, what are you going to measure through the nasal fluid?
Not nasal fluid, sir. Its sweat is there.
It's sweat. Okay. What are you going to measure through the sweat?
Whatever analytes we are measuring from this.
I think we already have ascertained that there's only one analyte which is very useful when monitoring, which is urease. Now that's related to kidney function. With most of these livestock, the problem is not with their kidneys. It's more to do with because they have got compound stomachs. So most of the problems are metabolic or endemic diseases. But it's a very good idea, and I'm glad that you've started thinking on those lines. And it's a very useful piece of information. I'm definitely going to go discuss this with Joe Neary and see if we can make use of this information. Thank you so much for that.
So I wanted to ask, what will be your insights for future? Like, hybrid is a thing, and genetically modified organisms are also made from cattle and all that. Like, new breed animal are there.
Well, as long as we at the end of the day, we are looking. For us, the genotype doesn't matter. We are looking at the phenotype, right? So as long as the function is the same, as the function is not altered, because what we are doing is we are having biomarkers. We're looking at biomarkers that represent a certain physiology that's taking place within the animal. As long as the physiology doesn't change, we should be fine.
And sir, I wanted to ask that how do you identify if the cattle is actually producing milk even after giving birth? Like, it is a milk producing cattle or not?
No, this one, we've worked on different cows. One of them was they call us beef cows. The other one was a milking cow. So what's your question? Sorry.
Why it is needed?
Why it's needed so milking cows is a much bigger need. As I said, we call them transition cows, right? Because between the lactation, once they've given birth to the cow, and between lactation, there's a period there where they're very much prone to diseases. That's where you have milk fever, mastitis, cow lameness, and stuff like that. I think you can speak to the to the gentleman who's got more experience here. But I think for the beef cow, it's more about the well-being. It's monitoring the glucose levels. Is it under stress? Monitoring lactate, monitoring ketones. because what happens is in UK we have the weather changes quite a lot. Sometimes we have high temperatures, and similar to humans in the UK, even the cows are not used to extreme heat, and they suffer a lot in the heat. So sometimes you need to know so that you can change the conditions, or you can bring in water sprinklers and stuff like that. So as I said, it's more about well-being. I mean, we are at the, wearable sensors is more now designed for well-being. We are trying to pull it more towards the clinical side of things, but for that we need to identify that it will actually make an impact there. So from the well-being point of view, the wearable technology is there. Again, we will only by doing some work and taking this wearable technologies more into the clinical side of things, by looking into ketones, hormones and stuff like that, we will know more about the clinical side of things. Infections.
Thank you.
You're welcome. I think my next talk is going to be on humans, so I think it might be better that if you have any questions, in the interest of time, we move to the next talk, as long as we've met the required criteria for this talk.
Okay.
Everybody happy with this talk? And the next talk is going to be on humans. So I just wanted to change this whole scenario. Again, just to mention, this is all unpublished work as well. You'll see some work coming out. We'll be communicating to a journal soon, so it'll be published in the next 2-3 months. But this is all unpublished results, so I'm very happy to share all of this unpublished result with you. Give me just 2 minutes to drink some water. No, if you have it, do you have it? I stopped recording, yeah. All right. So everybody, welcome back. I hope I've not tired you enough too much. So welcome back to this next lecture, which is part of the same GIAN series. This is basically on microneedle technology based subdermal diagnostics, right? What I want to do here is you'll be seeing a lot of slides which you might have seen in the past. I'm just going to take you back again. We're going to do mostly a recap of all the human studies that have been done using these subdermal devices, and I think it'll be a good recap, especially given the fact that we've just visited the animal studies there. Now, when we were talking about the animal studies, I did tell you about how useful the human studies have been and how we took the experience from the human studies into the animal studies, right? I was trying to, we were trying to look at the analogy there. We were trying to look at the different scenarios, what happens in ambulatory humans and what happens in ambulatory animals, how we can learn the lessons. For example, I mentioned about AI assisted devices, right? That was something that came from our experience of doing human studies. So these are the studies that have actually taken us there. Now, I would also like to mention one thing here, which is about digital twin. Have you all heard about digital twin? Right? So digital twin is a technology which kind of helps you, which is a twin, in the sense that it's analogous. There's some common goals there, and then you can learn from that particular technology. So one such technology is this technology, which we call as this is basically a commercially available device, Freestyle Libra. It's basically a continuous glucose monitoring sensor that monitors glucose, but this is not inserted, this is something that's implanted, right? So what I'm going to do is I'm going to open up this box and show you how this is used, and then I'm going to show you how this is a digital twin to the subdermal wearable technology and how we use this technology to develop our subdermal wearable patches, right? Okay, this is more used mainly for glucose, so we can learn from the experience of this particular technology, from the roadmap of this technology, the success and failures of this technology, as to how we can how we can develop things further as well for the subdermal patches as well. Okay, I'm going to open this. Has anybody seen this before? How many of you have seen this before? Libra 3, I'm talking about Libra 3, not Libra 2. Okay, so nobody's seen Libra 3. Okay, so these normally come with an app on your phone. You need to download the Freestyle Libra 3 app that you normally have, and then you have this particular device here. And this morning we had this lecture where we were talking about NFC and those kind of things. Freestyle Libra 2 works on NFC. This one is more, I think, Bluetooth based. So information that's generated by this sensor goes onto your phone directly, right? There was also this mention about using this as, having to run it past your, using NFC and doing a quick scan. That was there for a reason. Right? And that's why I say this is a good digital twin. The reason they started doing this was, you talk about implantable sensors, they are implanted quite deep into the body. When you implant something deep into the body, the problem you have is you will have biofouling effects, because don't forget, these sensors are meant to be used over a period of 14 days. Right? You implant something in your body subcutaneously for 14 days, there's definitely going to be some biofouling there. And what does biofouling do? It goes and contaminates, or it goes and basically forms a capsule of protein around your sensor surface, right? And that actually starts to affect your sensor performance. So what happens is you start getting problems with accuracy and precision of your sensors, and that in real life would lead to false alarms. There have been times when people, the sensors are reading low glucose values. Why? Because you have a capsule formation, there's a less amount of glucose actually diffusing to the sensor surface. And when you start reading low glucose values, that's an indication of a person going towards hypoglycemia. You start to get alarms, false alarms, right? And people look at it and say, I'm feeling fine, but my sensor reading is dropped down, right? So this used to happen when people used to monitor things continuously, always looking on what the readings are. So what people have started doing now, that's why the technology was modified, and they've started introducing something called as flash devices, wherein you take your reader and you flash it against your device, and you get the readings just when you feel that something is wrong with you. So you kind of use your own instinct, and you use what your instinctive feeling is, and you then look at the readings and say, oh, now I can confirm that there's something going on there. So that was flash technology. This technology is also similar, but here, as Professor Khalid mentioned, you're using a Bluetooth, so all the data goes through your Bluetooth. You need to keep your Bluetooth on in your phone and in your app, and it directly goes in there, and whenever you want to watch your readings, you can watch it there and then. Okay, so let's open this up. That's how it looks like, right? Yeah, that's how a Freestyle Libra 3 sensor looks like. Simple thing, you open it up, right? Ideally, I would advise you not to look into it, because if you look into it, what do you see? Can you see it on the screen? No, you can't see it on the screen. You can see there's a hollow needle in there. You would think, oh, this hollow needle, oh, this is, I'm going to get a hollow, you know, you don't want to, so you take this, you just open it up. This is where the hollow needle sets in there. You take this one, and then you can go at the back of your arm, ideally somewhere. It depends whether you want it visible or not. Some people, especially who are into sports, they like things to be visible. So you go in here, you can use your right arm, you fold your arm, and just, did you hear the click? That's because it's a spring-loaded applicator. I did feel a bit of, slight bit of a pain, a tingling sensation there as well, as you would call it. And then you can just take it off. The sensor has already been implanted, right? Got it? Ease of use, right? That's why I call it a digital twin. So now we need our subdermal wearable devices to have the same kind of easy format. You see, I did a demo and I showed you how you can use these sensors now. Okay, and then this can be disposed of, and the next thing you do is you bring your phone, and it says apply a sensor. It gives you all the directions, all the instructions that you need through the app. You say next, and then you scan your sensor. Should be scanning the sensor, and then it will start working in 1 hour. There's a warm-up time of 1 hour. There we go. And back. Next, scan the sensor. I'll just make sure that my Bluetooth is working so that it can pick up the sensors. Yes, the Bluetooth is on. Sorry. Scan new sensor. It should be fine. I think this. There might be some issues there, but that's more an issue with the Bluetooth there. But the reason I showed you this sensor, and the reason I showed you the whole implant, it'll come up. It'll come up soon. I think because we have to stick to the time of this presentation, so I'll move on to the presentation here. All right. So did you get the idea of digital twin? Did you get an idea as to how we use a digital twin to kind of develop our product of interest? You take an example and you say, oh, this is where I can where I can learn some important lessons, and which I can then adopt and implement for my own piece of technology, my own medical device that I'm planning to use. Okay, so this one you'll see a lot of repetition, a lot of slides that might be repeated, but that's again just to consolidate on the facts and to ensure that you've understood this whole idea of subdermal wearable sensors. Now, the nice thing about this sensor is the ease of use, right? I just mentioned. I'm going to run through this whole thing. So the learning objectives of this particular talk is basically to understand the role of microneedles in interstitial fluid access, how you can access the interstitial fluid. Let's review the biosensing device that's used with and using the electrochemical methods, and we'll also be exploring integration into wearable platforms, and then compare this subdermal with sweat-based sensing as well. Just basically a comparison between subdermal. This is going to be the last lecture, which I believe on the wearable side of things. After this, we'll be discussing other things which will be more challenges and stuff like that. But basics, fundamentals, and all those things that you need to venture into this field, this is going to be the last lecture for that. So please pay attention and make sure you make the most of this. So first of all, question that we ask is, why do we need subdermal diagnostics? Right? So what's the difference between epidermal and subdermal diagnostics? Well, if you look at this microanatomy of the skin, In case of epidermal sensors or epidermal wearable sensors, you have sweat that's on the surface of the skin, and then you try to, that's something that can easily be accessed, not a problem at all. But as long as you're sweating, you've got some sample there, so you access that and you can measure that, and you can measure that for thing. It's easily accessible. That's a nice thing here. Now, to access anything underneath the skin, you need to have something which is a bit more sharper, right? Something that's minimally invasive. To access the skin interstitial fluid, I showed you examples. You can use suction blisters, you can use microdialysis. That's more for clinical side of things. If you really need some biomarker which is in the interstitial fluid, but that actually has got a clinical outcome there. But for things like healthcare, where you're trying to monitor your metabolites of interest, you need something which is more minimally invasive. This was actually the main purpose of your continuous glucose monitoring sensors was more for clinical purposes, mainly to monitor your glucose in a continuous manner, so that that would help you administer insulin accordingly and manage a clinical condition which was diabetes. That's where it all started. Now they are expanding on the market. Now they're saying they want people to try these sensors, have different kinds of food, to understand your own physiology, what's going on in your own body, to have different kinds of alcohols, people who are big fans of alcohols, to see which alcohol gives you a spike, which alcohol doesn't give you a spike, which food gives you a spike, which food doesn't give you a spike. We have these YouTube videos from people using similar devices called as Zoe devices, wherein people have these devices and they have different types of foods. They have food, for example, just a normal naan with dal, and some people try naan, dal and some salads with it, and they show that when you have salads, your peaks are comparatively lower. You don't spike that much. So this is basically trying to work out your own lifestyle. This is basically wherein you're trying to see what is good for you. Basically personalizing your own diagnostic devices, or personalizing your own well-being by using these kind of devices there. So skin interstitial fluid, coming back to that, reflects what we call as the blood metabolite, because you have this subvascular plexus there, and the subvascular plexus is in continuous equilibration with the skin interstitial fluid. Skin, as I mentioned, is the largest organ in the body. Skin interstitial fluid, you have about 12.5 liters of skin interstitial fluid, and it is a treasure trove of biomarkers. And we've also seen this new hypothesis, this new reports which suggest that if you have two organs which have got the same embryonic origin, if they are from the same ectoderm, mesoderm or endoderm, then they would have the same form of biomarkers there. So skin is a good way, because skin originates from the ectoderm. Any other organ that originates from the ectoderm will have, you would be able to see the biomarkers similar to that particular organ there as well. Skin has got a lot of nerve endings. So if you're looking at neurodegeneration, you can actually look at skin. You can all have biomarkers of neurodegeneration as well. The skin interstitial fluid can be accessed in a minimally invasive manner, as can be seen from this particular image. I'm showing you this image again, but I think this is the best representation to show you the difference between a hypodermic needle and a microneedle. And also we'll explain you the fact that why we do not want to use the term microneedles, because if you say microneedles, people would think that this is needles, and then they imagine this kind of a needle here, right? But when you say subdermal wearable device, which have projections or micro spikes you were using, which was a term that we introduced initially, then it's much more acceptable. As I said, the devices are useless if they're not accepted by people. If you scare people right at the start by using the term microneedles, it then puts them off, all right? And then the devices don't get very high acceptability. And that's the problem that you've had with these. These have become very popular now, but earlier the acceptability was very, very poor. The acceptability was less than 20%. So less than 20% people with type 1 diabetes would actually carry on using these devices. Although the clinical evidence was that the more you use continuous glucose monitoring sensors, the more control you have on your glycated hemoglobin. Does everybody understand glycated hemoglobin here? So for those of you who don't know what glycated hemoglobin is, the blood which carries the sugar, right? The main component of the blood is hemoglobin, right? The main function of hemoglobin is to carry oxygen. So it binds with oxygen and it circulates oxygen all over through the entire body. However, when you start having sugar, and when your sugar levels start going much higher, then what happens is the high levels of sugar start binding to the hemoglobin as well. And when they start binding to the hemoglobin, then they form a compound which is called as glycated hemoglobin. So glycated hemoglobin is a good indicator of your disease. A lot of people who are suffering from type 1 or type 2 diabetes, the way because glycated hemoglobin tells you about your behavior over the last three days. You can have a glucose test next morning, you can do fasting, you can fast for 3 days and go and say, oh, my sugar levels are quite low there. That doesn't give you a good indication of what actually your glucose levels are. A good indication of your glucose levels is glycated hemoglobin, and that's what's normally measured either 3 months or 6 months. After a certain age, it's measured every 6 months, or sometimes even annually, right? Because that's telling you how good you've been, not over 2 days. You can do 2 days of fasting and eat a lot or the rest of the days. It gives you an average over 3 to 6 months, and that's a much better measure of the level of glucose level in your body. All right, so coming back to the ISF, you can tap into the ISF with minimally invasive microneedle devices, and you can access the biomarkers, and you can monitor things in real time. And the nice thing with ISF is also the fact that there's less variability with sweat, and because it's in direct contact with capillaries, you have direct access to capillaries as well here. As I mentioned, you have these subvascular plexus which will give you direct access there. Okay, I've already shown you these images before. Microneedles, for those of you who are attending this for the first time, microneedles can come in different shapes and sizes. You can have solid microneedles, hollow microneedles, and they can be fabricated using different techniques. This is not new. These have been there since 1970s. I think I might have a slide next. Yes, so they've been around since the 1970s. The original application of these microneedle devices was for transdermal therapeutic drug delivery, right? So they were mainly used because, as I said, the skin has got the stratum corneum layer, and the stratum corneum layer basically acts as a fantastic barrier, and it prevents the diffusion of that's what gives it the skin the property of being waterproof, right? And it also acts as a barrier for any foreign substances to go through the skin, right? So if you want to administer drugs through the skin, you have to break the stratum corneum. Otherwise, there are only 20 drugs that can be applied transdermally. The balms and all those things, those are all transdermal, but they don't go that far. They are more kind of placebo effects, because you normally smell the balm and you feel irritation in the eyes and nose and you think you have already been cured. That's the placebo effect there. Not all of that actually goes into your systemic circulation. Okay, so microneedles have been there since 1970s. As I mentioned, they come in different shapes and sizes, and the real applications were mainly drug, which is transdermal drug application or transdermal therapeutic drug administration, and most importantly, recently they have been used for intradermal vaccine delivery. And this was very, very important, especially when we had the pandemic, when the whole world was looking for a vaccine. Right, vaccines were available, thanks to the way we all worked very hard and got the vaccines right and this thing. But distribution of vaccine was a big problem because of the cold chain requirements. Right, if you can administer drugs or administer vaccines intradermally using microneedle arrays, you can actually then address the problem of cold chain, because the vaccines are normally in the form of powder, straight away loaded onto polymer films, and they go and dissolve themselves in the skin interstitial fluid. Because they're in solid state, you don't need any cold chain there, right? So that's why these microneedles are also very popular for therapeutic drug delivery applications as well. And you can actually combine them. You saw the talk on artificial pancreas, wherein you were talking about insulin pumps for delivery and microneedle array and your sensors for diagnostics. You can combine therapeutic drug delivery and diagnostics and make it into a theranostic device, wherein the thera comes from the therapeutic drug delivery and the gnostics comes from the diagnostics. Very, very useful tool for vaccine. You've administered a vaccine somewhere in Kampala or somewhere in Accra. You do not know whether the vaccine is efficacious or not. But if you have a diagnostic, if some of them have been modified into a sensor which can measure immunoglobulins, you know whether the vaccination has been efficacious or not. Similarly, cancer immunotherapy. You can basically administer the immunotherapy and then measure the response to the therapy by using the diagnostic tools. So these are some good theranostic applications. Coming back to the sensor side of things, coming back to the subdermal sensors, we normally have a standard setup. Your standard setup would be what you normally see in an electrochemical setup. You always have a 3 electrode system or a 2 electrode system. You can have a working electrode, a counter electrode, and a reference electrode. Right? You apply the potential between your working electrode and your counter electrode, and you have your reference electrode to keep the potential fixed. and you measure the current there. All right, electropolymerized phenols, and you can also have very thin layers. Now again, you saw me implanting this device, right, and you saw there was a needle there, right? Has the needle gone into me? No, the needle is pulled out. Why do we need that needle? Puncture it. What else? Why do you need a hollow needle if you were just going to puncture it? To insert the sensor. The sensor is in the form of, you had that CGM device, right? Have you got it right today? No. No, you don't have it. But yesterday, yeah, we can show you those. We can show you those. Basically, they have a filament, and those filaments are, you know, using the applicator. They're pushed in through a hollow needle, right? And the reason they use a hollow needle there is, number one, the needle allows you to puncture and go down subcutaneously deep into the skin, right? And the second thing it does is, your needle is actually coated with a membrane. It protects the sensing surface as well, right? Why? Imagine anything that you push into the skin. If the layers, because you're just imagine you have a blank device, you metalize it with a very thin layer, 50 nanometers, 100 nanometers of your metal. On top of it, you have a thick layer, right? What happens if you push the thick layer through the skin? It will be completely delaminated, right? So you need very thin conformal layers. You need to have the functionality of your bioreceptor maintained. And how can you maintain that? By having films that are conformal. For example, electropolymerized polyphenols, electropolymerized polypyrrole films. These are, the reason we use these are because these do not grow beyond 50 nanometers. I have done studies wherein I used epoxy polyurethane membrane, thinking that it will help me in reducing any interference. And I found that when I would push it into the skin, right, and take it back and do an SCM under the lab, the membrane would be completely torn, right? So we need electropolymerized polyphenols, electropolymerized polypyrrole films, because the electropolymerization ensures that the thickness is 50 nanometers and it's conformal as well. And then we need to functionalize the enzymes. We need it also entraps the enzymes there and keeps the enzyme fixed there. Otherwise, you need self-assembled monolayers to hold on to the enzymes. The enzymes are not very biocompatible if they leach into the skin compartment and through the skin compartment into the systemic circulation. If you look at the label of the enzymes, it says be careful, it may cause skin irritation. If it can cause irritation on top of the skin, you can imagine what it could do under the skin as well. So you've got to be very careful. So make sure that you have entrapped films there as well. This here is a cartoon showing you how a microneedle array looks like. You can see here you have a reference electrode which looks like a bit pinkish in color because it's a silver electrode which has been modified into a silver chloride reference electrode. Very easy to do that. You have this engineered lactate oxidase enzyme, because the problem with the commercially available lactate oxidase enzymes is you were not getting the dynamic range of 0 to 10 millimole concentration. And then you can see here you have a counter electrode which is made up of platinum, and then you have your working electrodes. These working electrodes are actually made up of platinum, and as you can see from those films on top of it, you also have those dots there, and those yellow dots are actually glucose oxidase which are entrapped in the film. Just to show you, everything is very conformal and nothing is sticking out and nothing can be delaminated when you push it into the skin, because the process of insertion into the skin is a mechanically, you're applying forces there, you're applying 10 newtons there. If there's going to be any deformity there, you can easily delaminate the layer, you can easily rip off the layer. And if the microneedles are not functionalized, how are you going to measure things? All right, I think I might have already shown you this picture. This is a picture from the first case study that we did, just to kind of do a quick recap. Again, look at the instrumentation here, right? See here, this is the potentiostat, a huge CHI potentiostat that we have, a device which has been inserted there. You can see we are still using crocodile clips there with wires coming out of the device. This was very, very crude, but for a first try, I think this back in 2013, this is something back 12 years back. I don't think this was a bad try. And then again, you need a gold standard there as well. Your gold standard is going to be one of the things that is normally used in the lab, in a clinical lab. That's your IV cannula here. The IV cannula is to draw venous blood. So you're drawing venous blood every half an hour, every 1 hour. You can't do that regularly, otherwise you can drain the person of the blood. So you're drawing blood every 30 minutes, every 1 hour, and then you're putting it into a machine, a YSI machine, which is used for measuring glucose in the venous blood. Right? And that machine is actually a gold standard. That machine is very well equipped to give you accurate and precise results. So as I said, your measurement, what are you measuring? You are measuring current. And what are you comparing that against? Glucose values. So you need to have a look at the plot, and you need those points there. If you remember from the animal studies, I showed you an enlarged view wherein you had the bolus and you saw a postprandial peak. You saw the capillary blood readings in between there, and by looking at the capillary blood readings going up and down and the trends that you see in your plots, you can see if you are able to capture the events. So what you try to do with your sensor initially is to capture the events related to boluses, the postprandial peaks, events related to exercise, the lactate peaks, where the lactate goes up, glucose goes down. So you try to capture the events there, and then you do later on bring in the AI and machine learning to make all of that make more sense. Again, as I said to you before, tolerability studies, which is a big challenge in animals. In humans, it's not a big challenge because you can have people there who can give you a visual assessment score. They can easily tell you, you're doing measurements in people who are over the age of 18, who can tell you clearly whether the device that has been inserted, what's the pain score for that compared to an IV cannula that has been inserted, right? So here you have a comparison of three things: the microneedle arrays, the cannula, and in this particular case you also have a CGM device there as well. And as you can see, the pain scores are normally lower for the microneedle arrays, which means that they are very well tolerated, right? If I would have been sitting here screaming that the device is painful, I think that is not a good sign here. The fact that I'm sitting here quietly and doing what I'm meant to be doing and forgetting that I have a device implanted means that basically I'm tolerating the device very well, right? Okay, and then looking at the instrumentation side of things, it's very important. I showed you in that image in the animal studies. If you had an instrument on a trolley, could you actually do animal studies? Can you have a trolley running around with a cow in the farm? No, you can't. And look at this here. So in phase 2, you have this young person holding a bag with the potentiostats in it. Can you give a cow a bag and ask it to carry it around? You can't, right? So you need something which is much more discreet, something which looks more like this PCB based device, which can hide under the collar, or you can tag it to the collar or something like that, which is light as well. You just imagine putting a 5 kilo potentiostat on the top of on the neck, around the neck of a cow. It will stop moving around, and then you can't say, oh, it's not tolerating it very well, because you've given it too much weight there. So you need devices which are not just low cost, but which are also convenient, right? I mean, this is an amazing example how convenient it is to implant these devices, right? When you talk about implantation, and these are invasive, I can feel that they have actually gone under my subcutaneous tissue there, and if I take it out, if it's hit one of my capillaries, I can even show you blood coming out there as well. But it's doing the job. It's sitting in there. There's no bleeding right now. There's no blood coming out, right? It's sitting in there and doing its job quietly, right? Convenience. I mean, cost-wise, it's very expensive, right? But again, because these devices are not perfect, that's why we are developing subdermal devices, right? Because these devices give a lot of biofouling. In 14 days, you would expect the biofouling effects to actually, when you pull out the sensor out, it comes the wire filament, which is normally bright in color, like bright white in color. When it comes out, it's gone all dark. And those of you who use the CGM sensor would know that when you pull it out, you can even see traces of blood there. You know that it's badly contaminated over 14 days. So what do? You bring in these subdermal devices. Why? Because these subdermal devices are not implanted subcutaneously. These are sitting on the surface. Skin interstitial fluid above the subvascular plexus, above the nerves, has got less amount of contaminants, has got less amount of things like your albumin proteins and things like that, right? What causes biofouling? It's the albumin, which is a bit lower down, higher levels of albumin. I'm not saying that there is no albumin at all. There is albumin, but it's reduced, and that reduced amount of albumin gives you reduced amount of biofouling. Okay, so this is an image just to show you about the instrumentation and that side of things as well, how the instrumentation evolved in the 3 years during the clinical studies we were doing. And then this is again to show you some data here. I think we've already had some talk on this about how you can smooth the data and how you can bring the AI side of things as well. But these are the early days. These are our results from phase 3 with participants with type 1 diabetes, where we did 24 hours monitoring, and this is how a continuous profile would look like. And then when you plot all of them without the AI, you can see that your Clark grid error looks very much scattered. If you compare that to the one that was shown this morning by Khalid, wherein you have that line in the middle here, ideally you would like everything to go stay along this particular line here. And look at the way the Clark error grid is designed. There is some margin of error more in the high concentration region. So this is your concentration, right? This is your concentration from 0 to about 400 milligrams per deciliter. This is the concentration, the range you have. It kind of goes from very broad towards, so you can see here, very, very broad, and it starts to kind of funnel in. It's like a funnel shape. And as you go towards a low concentration, which means that the margin of error is much higher in the high concentrations, but in lower concentrations you have to be very careful. And that's again bringing us to the same problem of hypoglycemia versus hyperglycemia. Hypoglycemia can put somebody into a coma. Hyperglycemia, I'm not saying it's not bad, you need to control your glucose levels, but that happens over a certain period of time. But when that hits you, that also hits you very bad. You might have heard about amputation of some people have infections is not being treated, you have to amputate the leg, right? These are severe cases of how type 1 or type 2 diabetes can go wrong. You might have heard about retinopathies, cardiomyopathy, nephropathy, all those pathies, they set in after a certain duration of hypoglycemia over a longer period of time, because all the hemoglobin is very busy binding to the sugar rather than binding to the oxygen, and instead of supplying oxygen to the cells, especially in the extremities, it's supplying them sugar. Sugar to cells is poison. Therefore you might have heard about diabetic people, when some of them, when they remove their shoes, the toes fall off because it all decays. It's like similar to frostbite. These are the kind of problems that you have, right? That's why this glucose diagnostics market is a big market, because you've got it's like an epidemic right now, the diabetes all over the world, especially type 2 diabetes, which we are ourselves responsible. I've been sitting on this chair for the last 4 hours just lecturing. I don't get to move at all. So we are all having a very sedentary lifestyle. You're all sitting and watching. I mean, I wish we could all move around, but okay, we can't do that. But of course, when you have the opportunity, then you should try to do some exercise as well. Okay, this is some of the results that we got from our Ministry of Defense project, where we were doing simultaneous glucose and lactate monitoring, again to show you how we had functionalized our glucose sensors for monitoring glucose and lactate sensors for monitoring lactate here. Again showing you the microanatomy of skin, and if you look at the subcutaneous tissue, if you have your the implanted sensors, they would go, if that wire goes down all the way down, deep down into your subcutaneous tissue, so it's quite deep below the dermis into the subcutaneous tissue, nearly close to the muscle. There are some results here to show you day 1, day 2 and day 3. I think I've shown you these results before as well, to show you what's happening after day 1. You can see the current values start to go down. Why is that happening? Because the sensor is degrading. Why is the sensor degrading? Yes, the enzyme is degrading, absolutely right. What is the other thing that's contributing towards the drop in the current values? So you have the sensor, you have the needle which is functionalized, biofouling. So you have a thick layer of protein being formed around this, which means that your glucose molecules now cannot diffuse easily because they have to go through this layer. So the amount of glucose molecules that can diffuse to the sensing layer, then that number goes down. Okay, so this is day 1, day 2 and day 3. This is some example of some lactate measurements that were done. As you can see, they've got a super fit athlete on the treadmill doing the exercise, and then you're measuring your lactate measurements in response to the exercise. Well, I'm not super fit, it's myself on the treadmill there. All right, and then this is the biaxial plot that I normally refer to. This is referring to the glucose. You have your current on one side and your glucose concentration on the other side. The blue plot is where you're measuring the current, and the green dots are where you have measuring your capillary blood glucose, and you have a lilac plot as well. That's your secondary reference. You can see that the trends are similar, so you can see that your capillary blood glucose value is going up, your blue plot is going up, and your lilac plot is also going up there as well. So all looks very good, and you're getting data which is, so as I said, what is important for us is these form of events, the postprandial events, where you can capture the events, because we are measuring current, and then you have to correlate the current to the glucose values, and for that there is an existing technology which is called as factory calibration, right? That factory calibration actually goes through all the enzymatic degradation process. It looks into the lot number. So all of these devices have got a lot number. They've got the day when they were all manufactured, expiry date and things like that. So you can get from that the batch of the enzyme that was used and things like that. You can use that, and then on top of that you use a lot of machine learning and build an AI model around it. And I think there are about 32 different factors that are used to build the whole the software that's used by Dexcom and companies like Abbott. Right? It also takes into account the biofouling effects as well. Again, because it's all predictive, the values you get are predicted values. They are still not accurate values. Okay, and then this also led us to this, bringing us back to the same point that I showed you during the animal studies, bringing in the AI model, right? And I think this has already been spoken a lot about in the last lecture that we had on AI and machine learning. Bringing in the AI makes your life much more easier. So for those of you who are planning to venture into wearable sensors, who would like to use even if it's physical sensors, please bring in machine learning and AI models to make more sense of your data, and do some correlation studies there as well, so that you can actually make more sense, and you can everything looks much more better there. All right, okay. And then these are the results that I already showed in the morning. These are day 1, day 2 and day 3 studies. The problem we have here, which I like in the animal studies and I don't like here, is these gaps that we have, right? This is because this is how the research ethics approvals, the Ministry of Defense ethics approvals were given. They did not want us to do any studies overnight, and that's why we had to basically stop measurement or stop recording during the night time. But anyway, the results look good with the AI thing. As you can see from the Clark grid errors, most of the green points and the lilac points are in zones A and B. We've all understood that. Everybody here understands Clark grid errors? So what are the 2 important zones in Clark grid errors? That's it. If you understood that, that's perfectly fine. And anything outside that either is an outlier or you need to optimize your device. And then what is this? What is BA? British Airways? Bland-Altman plots, right? So this is a Bland-Altman plot, which is basically a plot of the capillary patch data versus the mean lactate concentration, and this line is the important line here. If your points are concentrated along that, then you know that and then these are the red lines there, which is your 95% confidence level. As long as everything is there, anything outside that are your outliers, or that means that your sensor needs to be further stabilized. All right, I can see a lot of yawning going on. Apologies, it's not my choice to have it back to back, but please bear with me. We'll be done in the next few minutes. All right, okay, so these are the human results. So what are the ways in which we can mitigate the biofouling effects? Well, number 1, we are using polyphenols, and polyphenol to some extent is useful in protein adsorption, right? Subcutaneously implanted sensors have got wonderful membranes, and because they have that membranes, and because you're trying to push a wire, can you push a wire into the skin without any mechanical support? And especially if the wire is coated with an enzyme and coated with a membrane, right? So the membrane is there to minimize the biofouling effects. When you have membranes, the membrane has got pores, and those pores, I think we already heard this from Khalid yesterday, have got pores through which small molecules like glucose and lactate can diffuse through. But albumin, which is a larger protein molecule, which has got about 100 kDa, which is larger protein molecules, none of those can actually go through those particular pores. So it's very important to have this thing, and that's how you can prevent the biofouling effects. Having said all of that, you still will have some biofouling effects. You cannot completely eradicate any biofouling effects, because the body will keep on sending different forms of different forms of proteins around it. Okay, the biofouling, you can also improve this. Polyphenol also increases the one of the reasons we use polyphenol here is it also improves the signal stability over prolonged wear, and it's a hydrophilic surface which is also resistant to interstitial fluid clogging as well. That's how polyphenol layers help in mitigation of biofouling. And then when you look at the human trial results, we got a good correlation. I mean, I've already run through this. Lactate tracking in ambulatory subjects also looked to be very fine. Everything is within the the Bland-Altman is within the zone that we are looking for. Okay, instrumentation, I think we've had a lot of lectures on instrumentation as well, so we won't be covering any more instrumentation. We'll be having a workshop, and where we will look at how how we're going to be making glucose biosensors, but we won't be covering it anymore. What you need here, I think I showed you that instrumentation slide, if you remember, wherein basically as I said, it's much more convenient if you have devices on a PCB, so you can have a PCB which can have you can have a potentiostat on a PCB, you can build a potentiostat on a PCB, you learned that yesterday. You can have a microcontroller there with all the algorithms, with the AI model, which can then help you in filtering the data and giving you clean output values or clean or good predicted values as well, taking into account all the things that undergo, including biofouling as well. Okay, so in terms of instrumentation, it's very important to have low cost PCB platforms, right? If you had instruments, if you had a potentiostat that costed £5,000 in building, do you think you can sell it to a farmer? He wouldn't want to buy it, right? He would say, I would want something which is much more low cost. So you need low cost PCB platforms with integrated ADCs and BLEs, and they need to be powered by rechargeable coin cells, or if possible integrated biofuel cells. We saw that paper from Weigau's group, wherein they were using integrated biofuel cells which were based on the lactate that's present in the body. And you need it to be in the form of a compact form. Ideally, look at this, that's why I keep on giving this example. The instrumentation here is on the back of it in the form of a disc. That's the beauty of this device. I mean, these devices, if they've evolved over different generations, you've had generation 1, generation 2, generation 3. This is Freestyle Libra 3. Look how they've miniaturized it, and everything is in this particular disc here. We will show you tomorrow four of those devices that have been removed, and we will show you the tiny little components they have on the PCB that's in here. And this has also got the facility to store. So if I lose, so all the data that's in here is stored in, they've stored it in this device, and when it gets the next Bluetooth connection, it then connects to the Bluetooth as well. Up to 8 hours, right? So that's the beauty, and that's why the reason I did this now, I could have done this tomorrow at the workshop, but I did it now is so that I can put things in the right context, and I can explain you this whole concept of digital twin. What should be our motivation? What is our way forward? Where do we learn the way forward? If we are doing things for the first time, what is the example we can learn from? And I think for us, the CGM sensors are our digital twin. If you had, and these guys can learn from us. How can they learn from us? They can start using the same sensors, and they can start doing multi-analyte sensing. They can't do multi-analyte sensing with this format. You may need a wire which will have different parts functionalized with different materials, but that's going to be hard. So you may need 2 filaments, or you may need 3 filaments. How can they do multi-analyte sensing? That's going to be an interesting problem for them to solve. So with the digital twin, you can learn from each other. All right, so that's your instrumentation side of things. And then I think we already covered this design, and we've looked at the clinical applications. I've given you examples where we've used the subdermal wearable devices, not only in healthy volunteers, but also in participants with type 1 diabetes. Critical care and sepsis risks are very important. Lactate monitoring during sepsis is very, very important. And you can also use the lactate sensors for performance monitoring in athletes as well. And this brings me to the last slide, wherein we're going to have a little bit of an interaction here, a little bit of a quizzing that will go on. First thing that I'm going to ask you is, what do you think are the advantages of interstitial fluid over sweat? Or I can turn it around if you want. What are the advantages of sweat over interstitial fluid? I think we did that yesterday. So let's turn it around and let's ask the question, what are the advantages of interstitial fluid over sweat? Yes, please. There's one hand up. Yeah, go for it. You can take turns. That's fine. The ladies first is always good policy.
People would not like to sweat every time.
Good. Nice.
ISF volume is high. Not everybody sweats a lot, particularly if somebody has high body hairs.
Well done.
Taking sampling is difficult.
Yes. I mean, not difficult, it's minimally invasive, but it's not non-invasive. It's relatively difficult. Mike may.
So the ISF has got many more chemicals inside it, but the sweats, that doesn't have the things.
Well done. Any more? Go on. Share the mic, please.
It's similar to the blood chemistry, like the values.
Yes. So both of you have given the answers. You said many more chemicals, and you said it's similar to blood chemistry. What is the important message there? More biomarkers, and yes, please.
It correlates with the capillary blood.
Well done. There's a very good correlation with capillary blood and venous blood there. Any more?
And sir, I already said it because the sweat in the winter season we don't sweat sufficient. And
Yeah,
but
agree. Totally agree. So sweating is a problem during winter time. So it's sampling is easier, but what if you're not sampling at all? What if you're not sweating at all? How do you sample then? Unless you have a pioneer sitting in the audience who comes up with a new sampling technique.
Yeah, contamination may be also issue where the bacteria produces some lactate and then measurement may give.
That's why I use the term pioneer. That's a very good. It's a contamination on the skin, causing bringing in the exogenous products. That's another thing. Any more? I'm really encouraged by seeing this response.
Depends upon the external atmosphere, that atmospheric heat and all. So concentration of analyte in the sweat may changes.
Well done. The dynamics keeps on changing in sweat, right? While in the ISF it stays
So ISF, it's close relative to the blood.
blood and it stays constant. Yeah, by and large. It actually, so skin, one of the terms people use for skin is a window to the body. By looking into what's happening in the skin, you know what's happening in your body, right? That's why skin is called as a window to the body. Yes, please. Any other? All right, I think that's very well answered. I mean, I'm very happy, Khalid, that this workshop has been for you. It's definitely a big success. Everybody has given me everything that I expected in terms of the answers. So very well done. I think you've covered all the points here.
Credit to them actually.
Yeah, credit to them staying awake and listening to the whole lecture. What role does polyphenol play? There's a gentleman on the.
sir, polyphenol can act as a redox layer.
Well done. Yes, redox layer. Do you need a redox layer?
It is also mechanically stable.
Is polyphenol a redox layer?
Sir, polypyrrole is I don't know polyphenol, but I guessing it.
Polypyrrole is okay.
Is a
But do you need a redox layer?
For movement of ion more efficiently.
So you're talking about the thickness. So it's
Yes.
much more thin film. Okay, fine. Movement of ions, I will take that one. This one I'm doubtful. Movement of ions, I will take that one.
So it can prevent the biofouling
Yes, prevent biofouling.
because it resists the albumin development or not.
Yeah,
So
similar to a epoxy polyurethane membrane. There are micro.
the background, it reduces background current,
Reduces background. Why does it reduce background current?
forms a small, like thin layer of
Exactly.
polymer.
So it forms a thin layer, and polyphenol by itself is insulating. What else?
So is it due to the selective permeability of the polyphenolic membrane?
That's been answered here.
Yeah.
Good. Same permeability. That's same thing here.
Sir, it block protein adsorption.
That's been mentioned here. Prevents biofouling. Well done.
It can be selective for analytes.
Selective for analytes.
Yeah, like a selective membrane.
That's been mentioned again here. Movement of ions. Permeability. There's one answer I'm looking for. Think on the lines of mechanical. That's where I did this demonstration here. What stability? Stability of?
So when we insert inside the body, it
Yes.
gets stable.
So it forms conformal layers, which gives the device mechanical stability so that it doesn't rupture. As I mentioned, if you have anything on top of it which is very thick, there's every chance that you can delaminate it, you can destroy it. Polyphenol definitely helps in maintaining the conformal of this thing. All right, I think that's all the answers I was looking for. Okay, Clark grid error. How does a Clark grid error validate performance?
So we can know how well our sensor is working.
We can know how well our sensor is working. How?
So by knowing like it falls on the.
How do you plot a Clark grid error?
It was
How do you plot a Clark grid error?
conscious.
Concentration versus what? Concentration.
Blood.
Blood concentration? Yeah?
Accuracy over time.
Pardon?
Accuracy over time.
Accuracy over time. Is it accuracy over time?
I barely remember.
That was
the
in
case.
Khalid's lecture. Accuracy over time.
Oh,
What happens in Clark grid error? So if I asked you to make a Clark grid error, I'll draw it for you. Right, these are the different zones in there. What are you plotting here? Sorry, let's go one at a time. Please speak in the mic.
I think it's the blood reference value over here in the
Blood
y-axis
reference. Yeah,
and the concentration that we are getting.
concentration.
Blood reference.
Are you sure? This is your subdermal wearable device output value versus CSI or YSI, which is venous blood value. Right, so you're basically plotting the output of your sensors, because again, remind you, you are not measuring glucose, you are measuring current values, and those values are processed using machine learning and AI tools and calibration steps into an output value, and you are then plotting that output value and against the blood reference value, and you're looking at the difference. If there's no difference, they will all fall along the straight line. If there is difference, they start moving away from either in this direction or in this direction, depending on positive or negative difference. Start moving out. Right? All right. That's good. Is Clark grid error clear to everybody? All right. Why is it narrow here and wide here? Why is it narrow in this region and wide in this region?
Sensor works well for the low concentration.
Sorry?
It works well for low concentration.
Is that the reason why this is fun in the funnel shaped?
We have more margin of error in the wider concentration.
Repeat that answer in the mic, please.
We have more margin of error in the wider concentration, and then this
Than this.
less.
Why? Yes.
Hyperglycemia. and
Hypo versus hyper.
hyper.
Right? So if somebody's glucose levels are high for a certain period of time, it will affect them, but over a longer period of time. But if somebody is in the low concentration region, there could be some serious consequences there. Okay, I think that probably takes me to the end. I hope I have given you a good flavor of subdermal wearable devices and epidermal wearable devices. If for some reason you found me showing more energy towards subdermal wearable devices than epidermal, or if you see a bias there, it's for a simple reason. I've been working on subdermal wearable devices for the last 15 years. But having said that, I still maintain the stand that these technologies are complementary, so I wouldn't rule out another technology. So if there's an opportunity and if I can access things through the sweat, I would go for that, especially for babies. If I can do something using sweat, I would go for that. All right. So I hope I've kind of given you the basic thought process behind building a subdermal devices, and I hope this is good enough information to motivate you and to encourage you to look into this area and actually find your own applications or take this work further ahead and build on the strong foundations that you might have built through this workshop.
So yeah, just one last question from my side.
Yeah.
So definitely microneedles have a clear advantage over normal needles.
Yeah.
So why don't we see microneedle based products in the market right now?
That's because microneedles right now, as I said, they've been introduced in
Long time,
1970s.
yeah.
There's the first microneedle product that's going to come into the market is going to be a ketamine patch,
It's going to come.
clinical application.
Okay.
If you ask me another patch that's available in the market, nicotine patches.
Those are microneedle patches.
Those are very small structures there, but those are not exactly microneedle patches.
Yeah.
But ketamine patches are microneedle based patches.
What about sensors? Why don't we see?
It's early days.
Early days.
None of the sensors have gone from TRL stage 4 to have reached TRL stage 4. There's actually a new company that's come up, if you want to look for that, BioLink. This is by a student from Joe Wang's group.
BioLink.
BioLink, BIOLINK. That has been floated in the market. That's based on microneedles.
Right. Is it something to do with the manufacturing process, or no? It's just people.
It's
have
a mix
not
of
pushed
everything.
it?
It's a
Okay.
mix of all the regulatory hurdles. It's a mix of approvals from the MHRA and also the technologies as well.
Okay.
But there is definitely promise there, and I would strongly ask you to follow that company BioLink. They are coming up with these products,
Okay. Okay.
and actually that is good for us. It opens up the space for us to spin out a company, isn't it?
Yeah. Yeah. Just for the sake of discussion, just 2-3 minutes more. We need to extend the session.
How much do we need more?
Just 3-4 minutes.
Okay,
we
that's
have
fine.
3 more minutes.
Yeah.
And then we'll wrap up. So another question is, this applicator that you are designing, is it required for microneedles? For
Absolutely, absolutely.
Can you design it in-house? Is this
Yes.
designed
So
in-house?
this one I've got the design as well. If you want it, I can. So the applicator that
Be
we've
good
used
to-
here, this one,
Yeah.
is all in-house. In fact, now what we're trying to do is, this is the, it's like a disk, a PCB disk. I did show you some previous studies there where we spoke about interference that we see, which comes from the base if they are metallized, fully metallized. So we've started using PCB disks, and we have our acupuncture needles soldered onto those disks, and then we have the wires and everything there, and on the back of it is going to be similar to what we have. As I said, we are using our digital twin to see what are the good things and take those ideas from this digital twin and transfer them on here. So next time on the applicator you will have something which, as I showed here from the that was in the animal study, sorry, the video. The applicator will then, so that was, this applicator is for animal studies, but you can use similar applicator with lower forces and lower speeds in humans as well.
Okay.
But yeah, it's a very easy way of making applicators. You can get designs online on GitHub and all those things. You can look there. You can get designs for and even 3D. You've got these programs for 3D printing. So if you have a 3D printer, you can make your own applicator, and you can design the springs. You can adjust it yourself. You can do some studies yourself. You can order the springs, and you can have a spring-loaded applicator as and when required.
Okay. Thank you.
Yeah.
Like there is an applicator to insert the device or implant it. Do we need something to remove it? Like
I mean, this has to be in there for 14 days. Otherwise, I would have peeled it off and showed it to you. No, it's not for removing. I think your question is about, do you need another device to remove it? No. Even my mom could take it off when I implanted one in her. This is the device. This is the applicator. This is how it comes inbuilt.
and so the things that is inserted in cattle and all that.
In cattle, again, you don't need to. You normally have a adhesive, again, which you need to be very careful about the choice of adhesive. You can stick it into the animal. I think that's the problem we're going to have with animal with hair, especially horses and cows which have got thick hair.
Sir,
So is the force as well as the mechanism may change depending upon the type of the microneedle you are
No,
fabricating or making?
it will change on the type of the animal.
Type of the animal.
Yeah, because if you have horses, they have thin hair. The cows have got a much thicker fur. It depends on the animal. If it's a beef cow or if it's a milking cow. Normally, milking cows are more like as if you've waxed them. I mean, they still have hair, but it's very thin. Beef cow, they have got a very very thick thing. So it will depend on the type of the animal you're working on.
Sir, I have a little question on the mechanism. So, as we are applying the polyphenols a monolayer, so in the cells also ascorbic acids are present. We know that the polyphenols are also redox active. So now
Are polyphenols redox active?
yes sir, they are
100%?
redox active. Yes sir.
I thought they were insulating.
No sir, they are redox. I have personally tested them. So
But did you make sure that you've removed any phenol in there?
phenol.
So what happens is, have you done it when you test phenols?
Sir,
Did you
I
start with polyphenol or did you start with phenols?
no polyphenols means like we are going with the catechol, pyrogallols, gallic acids, tannic acids.
Okay, that's your definition of polyphenols. When I say polyphenols, what I meant is polyphenol as in the polyphenol that you obtain by the polymerization of the monomer phenol. You're talking about compounds that have got additional functional groups there.
Yes,
I'm talking about phenol when the monomer phenol C6H5OH, when you polymerize that, the polyphenol you get out of that is insulating in nature.
Okay, so that's a polymer.
That's a polymer of the phenol.
Okay.
But if you're talking about, if you something polyaniline, for example,
Yeah.
you have a different functional group there.
Yes.
You may have some redox activity there.
Yeah.
Okay. Or you mentioned about catechols.
Yes.
They may have some functional groups, OH groups and NH2
Yes,
groups.
yes.
You will have some functionality there. But the phenol I'm talking about is phenol, which is basically C6H5OH, which when on polymerization gives you polyphenol.
Another question, sir. We know that as from the Marcus theory also, the electrons can move beyond with a certain distance. Okay, so they can also move for the nanometer levels. So is there any possibility that the reducing agents inside the cell, they are reducing the engineered glucose oxidase? Because as sir told yesterday that they are engineered and the FAD moiety is like naked outside. So that's why the redox potential is being very high, 0.65 volt. So is there any chance, because we know that this intracellular reducing agents like glutathione, NADH, like ascorbates, they can, they are very fast moving their electrons towards like anything.
Yeah.
So are they going to alter any changes, like they can produce the FAD superradicals or like that?
That's actually a very good question. So what happens with the microneedles is it sits in the interstitial fluid which is between the cells. That doesn't mean that they would not puncture the cells. They will puncture the cells, and anything inside the cells that is your concern will be released, but the concentrations are going to be very, very low. Right? What you need to appreciate here is, what is the concentration, what is the dynamic range for glucose, everybody? What concentrations are we measuring of glucose? Sorry? 0 to 40 what? Millimoles. Compare millimoles. What you're talking about attomolar concentration. Attomolar meaning very, very low. So when you have something which is very low there, it's even below the baseline of your current.
I'm not asking about the competitive reactions.
Yeah.
I'm just asking that whether they are modifying the glucose oxidase.
They cannot, because that's what I'm trying to tell you. The concentration of those cellular products, intracellular products, is so low that the concentration outside is very high. It's like, there's a fire, there's a fire, forest fire, and you're taking a glass of water and trying to douse the fire off. You cannot, because the scales are different. One is a forest fire, the whole forest is under fire, and here is somebody who's just taking a tiny bit of, that's what I'm trying to say. So your intracellular components that leak out of the cell because they've been punctured by the microneedle, the concentrations are so low that they will not affect or they will not interfere. That makes sense?
I think that's all for today. If you have any, I don't think anybody has any more energy.
No, I'm sure. Let's let them have their
wrap
lunch.
it up. I think
They're sure they're all very hungry.
Thank you very much.
Really appreciate
for
your patience. Really appreciate you being here. I really appreciate it. This is something which would have been virtually impossible in the UK. to get students to skip their lunchtime and attend lectures. Thank you so much, everybody.
Thank you. Thank you to you also.
It's thanks to you wonderful people that it motivates me to teach as well. Thank you.
So that's all for today. So we will resume tomorrow. So sir has only one lecture left. So actually in the whole series there is only one